to introduce you to our Dean of our Faculty of Life Sciences and Medicine, Professor Ajay Shah, who will say a few words before the symposium starts. Ajay, yes, you're live. Thank you.
Good afternoon, ladies and gentlemen, colleagues. It's my very great pleasure to welcome you to this international symposium on the role of metals in brain health and disease. We have a fantastic program that the organizers have put together for us, and like you, I am very much looking forward to it. Now, I do not need really to say much about the fundamental role that metals play in biological processes to this audience. Being a little bit interested in that area myself, perhaps just to note that from an evolutionary perspective, how fundamental is the role that metals play, going right back to the earliest origins of life, redox activity of metals, their interactions with oxygen in key biochemical reactions, contributions to disease. So really fundamental. It seems to me that the focus of this symposium is also really opposite, because focus on brain disease and dementia addresses one of the major unsolved problems, challenges that we face as a society. So I really would like to congratulate the organizers, the London Metallomics facility and the Physiological Oxygen Laboratory for putting together this wonderful symposium. And I would say that it really exemplifies for me some of the USPs of King's College London and our faculty and our ecosystem. So we see this highly productive interaction, intersection perhaps, between metallomic analysis, chemistry, imaging, biology, disease. We see an intersection between the lab and the clinic. We see the intersection of technologies, and we see the intersection between different faculties in our institution, all focused on a common challenge. So that interdisciplinarity and that collaboration that Giovanni and other colleagues who are involved in not just the symposium, but the underpinning activity that, frankly, the symposium is supposed to showcase, are to be congratulated to maximally leveraging the potential that we have in this institution. So let me just end by thanking you all again for attending, welcoming you to the symposium, and let's look forward to some wonderful science. Thank you all very much.
So, sorry, I've put my mic on. I presume we now are going to move to Wolfgang Marit as the chair. Please, Wolfgang.
Yes, thank you, Giovanni, and thank you, Dean Shah, for this wonderful introduction and taking the time to be with us. Just a few words. Metals at King's College has been a theme in pharmaceutical sciences and nutritional sciences for almost 30 years. And as a result of that, we have a group of people, it's called the London Metallomics Consortium, which put a Wellcome Trust together many years ago. And that was the start of the London Metallomics facility. And without pointing out all the participants, I want to thank Thea Stewart for doing a wonderful job and develop that over the last years. Phil Flower was instrumental, Bob Hyder, and many others. And I also want to mention that King's College was instrumental in developing this. grant further, which was actually with Imperial College. Namely, they put a lot of money in, also through funding us through a grant which is called Kings Together, which allowed us to build this facility over the years. So with much further to do, I would like to introduce Thea Stewart, who now moved on. She was the manager of the facility to a position in business development of the core facilities. And Alex Morel is now the manager of the facility, and they will both speaking about the opportunities and the recent developments in that facility. So Thea and Alex is all yours. Thank you very much.
Great. Thanks, Wolfgang. So if you can all hear me, I think we're good. I'm just going to share my screen. Okay. And I think that should be good. Does everyone see that? Yeah, perfect. I'll assume yes. Great, so I just want to say it's a real pleasure to be hosting the second Metallomics Symposium this year. The theme is obviously the role of metals in the brain. And those of you who have seen me present before, some of this is going to be familiar, but for those of you who haven't, I'm going to be presenting a bit of an overview of the field of metallomics itself in general and the LMF. And basically, I hope to show all of you how significant this area is in really a multitude of disciplines and how the LMF is uniquely poised with the cutting edge analytics that we're developing to underpin the complex questions, including the roles of metals of brain health and disease. So as Wolfgang mentioned, I moved into a business development manager role. And so I'll be handing over the presentation halfway through to the new LMF lead scientist and manager, Dr. Alex Morel. And he'll be speaking a bit about the trajectory of the LMF moving forward. And he's going to be presenting some very exciting developments that he'll be bringing with his expertise. So without further ado, presentation. So I always start my presentations stripped down, fundamental building blocks of life, right? So we're all familiar with this periodic table, but I think it's something that we take for granted. And really through the process of developing the London Metallomics facility and getting more steeped in the field of metallomics, I've come to a new appreciation of this table. And it's really through countless interactions between these elements, we have life as we know it. But I think what most of us don't realize is that 85% of the periodic table are actually considered metals or metalloids. And although these metals only make up 2.5% of the human body, they're absolutely as important as the other 97.5%. So simply put, really no metals, no life. And underpinning this life are the complex roles of metals that are really inexplicably intertwined in every single biological function that spans from the genome to the metabolome. And so this is why I often refer to metallomics as a master integrator. So what are some of the questions that we ask in metallomics? Well, we're wanting to understand the local coordination environment around an element. We're wanting to understand the types of ligands that are bound to that element. We want to understand structural and functional roles of metals within biomolecules, their subcellular tissue or whole body localization, and even the dynamics within these processes. But ultimately, what we're trying to answer is the role of metal chemistry dynamics in biological processes within the context of a complete and whole and functioning system. So we already know quite a bit of the significance that metals play in biological processes, but it's really just the tip of the iceberg. And I think after hearing all of the talks today, you'll come to appreciate that there's a lot more that is still to be known and still to be studied when it comes to metals and specifically brain health. So a little bit on LMF, we've been focusing the last 4 years to really develop a cornerstone of elemental bioimaging. And so I'm just going to show a few examples. So we can image in different spatial scales from cells to tissues. So on the left-hand side, you see an image of a brain, of course, for today, and the relative distribution of zinc, copper, and manganese in this case. But we can also measure at cellular resolution. So on the right-hand side, you see the zinc distribution within mouse brain endothelial cells. What's very exciting now is we're starting to incorporate some new capabilities. So using the latest technology from laser ablation from Teledyne and coupled to ICP time of flight from new instruments, we were able to have a demo of this new capability, which allows us to capture almost the entire periodic table simultaneously. And so this is just a brief example of some work that was done down in Southampton on this coupled system, showing all these isotopes from a wild type brain that we looked at. And this is powerful because what we can do is we can image the endogenous metals, but we can also start to image exogenous metals, specifically metal tagged antibodies, and what we call a multiplexing approach. And Alex will get into this a little bit more detail in his half of the talk. We can also look at everything from elements to small molecules. So this is an example of some work that we did with the zebrafish embryo, looking at lead uptake in the brain. And you can see here on the left, the zebrafish embryo, and in red, the lead uptake. And this is just proof of concept, but we were curious, can we start to try to correlate the elemental bioimaging with other imaging modalities? In this case, we went to Imperial College with Sarah Fern, and we did some secondary ion mass spec, otherwise known as SIMS. And what we picked up on, interestingly, was a metabolite ion called leucodopochrome, and this kind of localized in the same region that we saw the lead uptake. So again, just proof of concept. We didn't have any kind of working hypothesis in this case, but just showing that the potential is there for this type of multimodal imaging approaches. And we can start to look at lipid signatures. So this is an example of an overlay of a specific lipid composition profile overlaid on a copper distribution in the brain. And by being able to use a technique called DESI, we can get lipid profiles and image their relative distribution to specific metals. And this is really helping us to explore the links between altered metal and lipid metabolism and disease. And I think you'll probably be hearing a little bit more about this in Giovanni's talk later this afternoon. And we can start to approach whole body applications, specifically with PET metallomics. That was really brought to the forefront by the work that Phil Blower has done at St. Thomas's for looking at the whole body metal distribution in vivo. And Phil is also giving a talk later, so he'll talk more about this. But some examples are imaging copper homeostasis in cancer, looking at zinc and manganese trafficking, anti-cancer trafficking, and I believe Phil will be specifically looking at its applications within the context of questions in the brain. So as scientists, we all know that there's absolutely no silver bullet. There's not just one application. But I often thought of in the past years, do we even know how many bullets exist? So many of you know that I wrote this perspective piece and coined this term multi-dimensional metallomic imaging, or an MMI approach, really trying to cherry pick and understand what the best imaging techniques are for metallomics questions that span whole body to subcellular applications. And what I quickly realized is that we have a lot of these techniques within the London Metallomics facility, within King's College, and also London. And I think the LMF is really starting to become an analytical platform that can start to combine a lot of these techniques in a correlative approach and take this MMI approach, as I coined a few years ago. So the LMF is developing essentially into a hub for comprehensive integration, a state-of-the-art metallomic analytics, trying to foster direct interactions between academia, industry, and instrument manufacturers, whilst providing the training, education, and expertise to support all of its users in the emerging field of metallomics. And with that, I'm going to hand over the presentation to Alex, and he's going to talk a little bit about the trajectory of the LMF coming up in the next couple of years. So I will stop sharing my screen and hand it over to Alex.
Let me just have a little look, see what I can do. Is that the right screen? That is the question. Can you all see that? That is the question. Wait, I can't see anything head, so I assume it's okay.
Yeah.
Okay, cool. Thank you, Sarah, for giving a really great overview of metallomics and really sort of emphasizing some of the areas where it's been especially important in the context of brain. I'm going to give you a brief overview of an avenue that we really kind of want to push and develop at the LMF, and hope it will become sort of more routine for our users, which is developing correlative imaging approaches. But kind of before I get onto that, I'll just kind of skim over our current capabilities just to, if anyone doesn't know them, or just to refresh them. So we analyze a variety of different samples, so from soft and hard tissues to dinosaurs. We have particular expertise in preparing and analyzing biological materials, but we do have the capacity to cater for a huge range of different types of samples. In terms of actual preparation of these samples for analysis, we have the facilities in-house and expertise for sort of digestion, including microwave, oven and chemical. And importantly, these are all done in a very ultra clean, metal-free environment. For solution-based work, we're often just given bulk samples, which we'll then process accordingly and have the facilities to load these into our instruments in an automated fashion, which means we don't have to kind of manually put 300 samples into the ICP-MS, which is great. We got the expertise and can offer advice on tissue preparation to generate tissue sections. And in the future, we're sort of exploring the idea of offering this as a routine service as well. Once the samples are actually prepared, so we offer two main routes of analysis, which are either solution-based or imaging. So for solution, we have a conventional ICP-MS. This is isotopically sensitive. And depending on the element that you're actually interested in and the matrix that kind of surrounds the element, we can step from picograms to nanograms per liter, and we can do this in a completely quantitative manner. We also have the ability to discriminate speciation using HPLC columns, and now can perform TXRF, which is particularly useful on low volume samples. And we have the equipment, and we're sort of testing and going through single cell ICP-MS now. The second, and arguably the more interesting route for me, is imaging, and this is where we have a laser ablation system coupled to a triple quad ICP-MS. So this kind of coupled system allows us to selectively ablate material which is introduced into the ICP-MS, which generates these like sort of beautiful images that Thea has just shown in her talk. We now have fully quantitative pipelines, which I'm very, very pleased with, and we're routinely imaging samples in the resolution ranges from 1 microns to 50 microns. So as Thea just showed in that brain, we image whole tissue sections, but also can go down to that cellular resolution as well. We now have just been got a new 193 nanometer Teledyne Iridia laser system. So it's installed very recently, and this has the capacity to run at 1000 hertz. This means it actually shoots 1000 laser shots a second. So with this, we have the capacity to image samples much faster than we ever have before. And in comparison to other elemental imaging modalities, we are a lot faster. So you can start seeing we can get through many samples to make it more better for projects, for sort of thing. So back to kind of the bulk of the presentation here. You may have heard of this buzzword going around called multiplexing, and Thea's already mentioned it today. And it actually originally comes from computing and telecommunications, where several analog or digital signals are combined to generate a significant advantage. And we use it in imaging, and it's used to describe the combination of more than one imaging technique. And this is to investigate a single sample. So to kind of split this up, you could think of these could be a combination of microscopy techniques, spectroscopy, spectrometry, or many more. And within these just broad subcategories, there's hundreds of different types of imaging methods. You'd be happy to know I'm definitely not going through each one of these. And this slide is just to really emphasize that there's such a small proportion of scientific methods out there that you can use to gather information about in this case, about your tissue sample or your cell samples, and you can use them to gain significant advantage of just using one of them. So I'm going to give you a couple of examples where sort of this multiplexing has been really useful. So here's an example where we can use a particular technique called nano-XRF to reveal the elemental distribution. And this technique is quite similar to LA-ICP-MS, gives the same sort of information. But this technique only gives us information about the concentration and the distribution of elements that you can see in the top image there. In this case, it's a distribution of cobalt and chromium from a hip replacement. But this tells us what elements there are, but it doesn't tell you about the actual chemistry of these elements. That's where a multimodal approach can be very useful, as once we've identified actually where the metals are in the sample, we can then use a targeted approach to reveal the chemistry of, say, chromium in this case, to see whether it's an oxide or a phosphate, And this is really important in these sorts of projects to work out whether they're cytotoxic or not. Thay has already shown some of the images here, but recently we've done a project with Professor Mann. We've been doing some correlative work with murine brains, capturing the elemental distribution using our LA-ICP-MS system, and the lipidomic profile with DESI-MS with key collaborators. And just to stress that these two imaging techniques was done on exactly the same tissue section. I'm not going to go into too much detail here. I'm sure you'll hear about it more later. But I just wanted to kind of outline here that we're starting to perform routine measurements at the LMF using multiple modalities, including proteomics, lipidomics, and metallomics, and we want to perform the appropriate analysis to actually correlate these types of techniques. And a big one for us is histology. So histology and immunohistochemistry is vastly used in fundamental science and in its routine use in clinical diagnostics. With the ability that I've just mentioned with the new Iridia, we have an improvement in our ability to perform elemental imaging so much quicker. So there is now a huge demand for correlative approaches between histology and elemental imaging. These approaches kind of can allow researchers to understand spatially the interactions between, in this case, exogenous but also endogenous metals within our tissues with specific biological epitopes, which is very powerful. In the case that you can see on your screen here, we can see the hyperaccumulation of titanium from a failed metallic implant, and that we can see that with the elemental imaging approach. And also on the left, you can see the histology, and you can see that there's cellular infiltrates that are co-locating locally at certain concentrations. And this is kind of suggesting that the titanium we can see here, stimuli, is actually pro-inflammatory. So kind of just to run you through how this type of, these images you can see here, how this data is actually done from the bottom up. Usually, sort of conventional route for preparation would be to acquire a tissue block, either formalin fixed, paraffin embedded, or frozen tissues. We would then perform some sort of sectioning to generate serial sections. And then these sections can then be analysed. As most of these imaging modalities have very specific requirements on sample preparations, or even the substrates, actually what the tissue is actually put on, are very, very unique. Immediately adjacent serial sections are often used for best representation. Now, the problem with serial sections is there is inherent variation in between adjacent slices. So if I play this video here, I'm very hoping it plays. This is a 30 serial sections just playing through it. And you can see that the tissue microanatomy is slightly different from one slice to the next. So whilst the sort of the large cellular infiltrates, which you can see in sort of the dark purpley blue colours, remain roughly in the same area, if you actually look at the cellular level, so smaller features, there's huge variation in between adjacent slices. So at this point, you have to really ask yourself whether the approach of using these serial sections is appropriate. So if you are looking at sort of the tissue level, so you're looking at large features, for example, the inflammatory infiltrates, which are highlighted in red here, these features persist through several serial sections. So if you're comparing the position of these in comparison from this one to an elemental approach, then you'd be fairly confident with the analysis and you'd be absolutely fine doing that. However, if you are now trying to look at the cellular level, so for example, you're trying to correlate a specific cell to another cell, or you're trying to look at a cell in comparison to a metal, as shown here, this really needs to be done on the same tissue section, as there's way too much variation in between one slice to another to perform any sort of correlative analysis where you want to quantify sort of the relationship between images or things. So we have this kind of task in hand. So to avoid these uncertainties and to create more powerful correlative approaches, we need to develop methods to image the endogenous and the exogenous, or the whole periodic table as they are shown, at the same time as imaging the tissue microanatomy on the same sample, importantly for these correlative approaches. And by tissue microanatomy, this could be just a type of cell, it could be a protein or architectural features within your tissues. So to do this, we need to label the tissues in the correct place with something which is actually detectable by our elemental imaging approach. I'm going to focus kind of on two potential solutions here, one being a metal conjugated with an antibody instead of a dye or a fluorophore, or using metal complexes directly, which have a natural affinity to certain biological structures. For example, the one here shown on the right, this ruthenium complex, binds to mitochondria in cells. Firstly, to go back to how conventional confocal microscopes work, these rely on antibodies which are conjugated to a fluorophore, which emits a specific color of light when actually imaged. And this imaging technique can produce beautiful images, which are shown here on the left, which are highly resolved. But a major advantage of using a metal tag instead of a conventional fluorophore is that there is significant spectral overlap, as you can see on the right, with emission wavelengths, which actually means in reality only a few antibodies can be used at the same time. It's also complicated by the excitation of fluorophores, meaning actually in reality only 4 to 6 simultaneous epitopes is kind of your limit for imaging. Antibodies which are specifically bound to lanthanides is not actually a new technology at all, and it's been around for decades. So they're originally developed for mass cytometry, but more recently they've been used for imaging purposes and have attracted a lot of attention in journals with images such as these ones, and they're being published in sort of Nature and Science, which is fantastic. And I've included Fluidigm here, and this is just a company who sells these reagents, but also has their own imaging platform as well. For metal tagged antibodies, Fluidigm offers a range of antibodies conjugated to different lanthanides. And as our LA-ICP-MS technique is isotopically sensitive, and some of these lanthanides have several isotopes, for example, neodymium has 7 different types of isotopes, there is a possibility of simultaneously tagging up to 100 markers at the same time. Now, there are some difficulties and limitations, but there have been several reports, including this one shown in Nature Methods, who have successfully tagged over 30 antibodies and have imaged them simultaneously. So to show actually this actually has worked for us, this is an example of some work we did where we managed to stain 7 antibodies and an intercalator for DNA. And at the same time, we will be able to image the exogenous material, which is in this case was platinum distribution, because this was a patient treated with cisplatin for chemo reasons. So with the 1 micron resolution laser, we could image the nuclei very easily and extract information about cells, positions, size, etc. We could then use a Ki-67 antibody to identify which cells are dividing, which in this case was really particularly useful for the cancer study. But at the same time, we can measure key architectural markers, including smooth muscles, collagen, and these markers are really important in our study because we were interested in how the rapid growth of cancer cells can change the extracellular matrix. And then finally, we can see where the actual therapeutic drug has accumulated due to that elemental sensitivity of the instrument. And at this point, you could then do some very clever analysis, do some nearest neighborhood analysis to see whether this accumulation of the exogenous material, the thing that you've introduced, is accumulating in the correct place. So kind of to sum up, we anticipate what we want to anticipate at the LMF, and that is to develop these multimodal imaging platforms so they become more routine measurement strategies for our users. And this could be including metallomic, lipidomic, but also the histological features within the tissues as shown here today. And of course, we would love to give a comprehensive level of support, analysis, and interpretation, as often you see when people do these techniques separately, the analysis between them and combining them is very difficult. And that's where we want to be able to kind of give a comprehensive level of support here, as we want to make sure everyone makes full use of what data they actually record. There's a lot of people to thank here from both Thea and I, starting with the LMF team, Alex and Renato, who are absolutely fantastic, the whole team at Teledyne for their continued support with the lasering, Thibault at Ghent and the entire Metallomic committee at King's for their valuable scientific input, the team at Thermo for the support with the ICP-MS, and Phil Shaw at Amtech for the collaborative use of the Vitesse system. Also like to thank Wellcome for the original funding for the LMF, and thank you all for being here and listening here today. Thank you.
Well, I guess I'm live. Thank you so much for this wonderful introduction. I think we are not taking any question here, are we?
No, we're asking people to put questions in the question and answer tab.
Good. Fine. So, and thank you for staying in time. I will make a short introduction for the next speaker, and then we're pretty much on schedule. Yes, I have to, unfortunately, another international meeting for just 1 hour, so I will not be able to introduce my colleague Phil Blower. And Phil, my apologies. So somebody else will jump in, but I think you all can live without that. So the next speaker will be Professor Doug Arslan from King's College. He's at the Institute of Psychiatry, Psychology, and Neuroscience. So we will now come to this focus on the brain of the entire symposium, and we are really pleased that he accepted. Everybody is very, very busy, of course, and it took a lot of effort and time to put this together. And Giovanni, thank you very much to you too, to putting it together. So Professor Arslan's talk will be Parkinson's disease associated cognitive impairment. And with that, I give the microphone, so to speak, to Professor Arslan.
Thank you, Professor Marek, for the kind introduction. Thank you, Giovanni, for inviting me. So let me first start by sharing my slide. Does it work?
Yes, it's working.
Excellent. Let's see, it doesn't moves. So thank you again, Giovanni, for inviting me. I told you I know next to nothing about metals. Know a little bit about brain, and particularly I know about people with brain diseases. So when I look at this amazing faculty of presenters, I feel like I'm a big fan of Hans Christian Andersen, the famous Danish author, and I feel like the ugly duckling here. But then on the other hand, I represent the people, the patients, who then hopefully in the end will benefit from the amazing work that you guys do with these fantastic instruments that we've just seen. So I hope that therefore it might be relevant also, and then show some information from the, let's say, from the patient focused. And my topic will then be Parkinson's disease associated cognitive impairment, and the reason for choosing that, in addition obviously to be one of my main topics, is that it will then inform you. I can see many people here working on Alzheimer's and many working on Parkinson's disease. It's important to be aware of that Parkinson's disease, as the other neurodegenerative diseases, they have a variety of brain lesions, a variety of brain structures involved, and a variety of clinical presentations. So whereas Alzheimer's disease is a memory disorder mainly, and Parkinson's disease is a motor disorder, they also come with a lot of other additional symptoms, and that will be the focus for this talk in Parkinson's disease. So I will start by debunking 4 myths about Parkinson's disease. First, Parkinson's disease is a nigrostriatal disorder. Yes, it is true, but in addition, it is a multi-system disorder involving a number of other important areas, such as the neocortex, even the thalamus, hippocampus, amygdala, brainstem, etc., and even extra cranial areas as well. And second one, Parkinson is a dopaminergic deficit disorder. Yes, true, but in addition, it's also a disease of other very important neurotransmitter systems: cholinergic, noradrenergic, serotonergic, glutamatergic, and other deficits are also common and play a major role in the disease. And then thirdly, Parkinson is a motor disorder. Yes, but it's also has a number of non-motor features, where dementia is probably one of the most important ones. And finally, Parkinson is an alpha-synucleinopathy. Yes, but also other protein lesions are involved, including the amyloid and tau and other pathologies, including inflammation, oxidative stress, mitochondria, autophagy, etc., etc. So Parkinson's disease is a very good example of the complexity of these neurodegenerative diseases. So before we start, we're just moving over to our dear Lewy body, where alpha-synuclein is a key protein, and in these diseases, Parkinson has been a model disease of alpha-synucleinopathies. The alpha-synuclein, which has a role in synaptic plasticity and transport, for a variety of reasons, start to fibril and aggregate, and then ending up with these intraneuronal Lewy bodies that can be visualized using a variety of techniques. And as you can see here, these Lewy bodies are present not only in the classical brainstem in Parkinson's disease, but also in other areas, as I just mentioned, including hippocampus, etc. So Heiko Braak has been extremely influential in outlining not only the pathology of Parkinson's disease and related Lewy body diseases, but also the progression of pathology. And this is created this famous Braak staging system. And the reason for me to show it is to again repeat or underline the importance of Parkinson's disease being not only a negostriatal disorder. You can see here that, yes, the substantia nigra is involved and the dopaminergic negostriatal pathways, but in addition, a number of other key brain structures are involved, both downstream and also upstream from the substantia nigra. You can see structures like the limbic amygdala, thalamus, hippocampus, and again the neocortex. And obviously the different patients have vary tremendously in terms of the degree of involvement of these different pathologies. So although we talk about Parkinson's disease, and it's the same with Alzheimer's disease, these diseases are extremely heterogeneous, and we could maybe better talk about Parkinson's diseases or Alzheimer's diseases, because the patients come up with a number of variation. For example, as we will talk about cognitive impairment, it's common, but not every patient, and at different stages develop cognitive impairment, and it's the same disease. So what are the features and the factors that lead to different clinical presentations? How can we predict this? And even more importantly, how can we prevent this progression taking place? And maybe work on the metals and related work can contribute to this. I will sneak in a little bit of metals in a couple of slides, but mainly keep a focus on the clinical and pathological aspects. So let's now move the focus from the pathology to the clinic. And this is a little bit a chaotic slide, but it demonstrates the frequency of cognitive impairment and dementia. To the left, you see a population-based cohort from Norway many years ago, and the mini mental state examination. I guess you have heard of this very frequently used crude global cognitive grading scale with scores between 0 and 30. And the point here is to show that in this community-based sample of 260 patients, we have the full spectrum of this scale. We have majority scoring fairly normal here in the range between 25 and 30, but we can see that we have all forms of scores, including down to 0. So we have a full spectrum of normal cognition, we have early cognitive impairment, mild dementia, moderate, and even severe dementia in Parkinson's disease. And importantly, longitudinal studies like this one from Sydney have repeatedly demonstrated that people with Parkinson's disease who survive, nearly all of them will develop dementia. This is then a 20-year study from Sydney, and we can see the curve here. This is psychosis and dementia with the full line. We can see that with time, everyone has developed dementia. You can say, well, if we live until we are 110, each of us will also have dementia, but the risk for dementia is much higher than in people without Parkinson's disease. So down here we can see, if you look at point prevalence in Parkinson's patients, we see that, well, half of them have normal cognition, 20% have dementia, and 25% have mild cognitive impairment, which is a pre-dementia syndrome, where they are already on track to develop dementia, but have not yet reached that track. We just published a review of the cognitive aspects of Parkinson's disease, and you can see some of these elements that we discussed in that paper here. And one of the key observations from work is that there's this huge variation. You can see some people develop early and rapidly cognitive impairment and dementia early on in disease, whereas others have a more slower cognitive decline, and some patients even remain with normal cognition for decades. And we also see this line, so it's not always a straight progression, but patients tend to vary and can even improve cognition, but then also tend to go back to worsening again. So in addition to being common, it's very important to be aware of that the non-motor aspects of, for example, psychiatric and cognitive aspects have tremendous relevance for the patients and the carers. So yes, it's important to focus on tremors and slow movements, but all patients today who are well educated, they are aware of their risk for dementia, and when dementia develops, it's a devastating stage of disease for the patients themselves, and obviously for the carer and for society. So we can see here the family burden, the carer burden obviously increases, and the functional impairment increases, and the risk for institutionalization and early death is increased. So these represent an extremely important and also very costly part of the disease, and that's why it's important to focus also on that. And I'm mentioning this here because the brain pathology that leads to the motor symptoms also contributes to the motor symptoms, just different regions involved. And here's one important work from the group in Cambridge, discussing the different brain structures and brain and pathways involved in different cognitive symptoms. So it's not a one size fits all. The cognitive impairment spectrum of Parkinson's disease is highly varied. We have some more frontal type, executive attention type impairment, which seems to be related. This is still a theory, but a very important model. Seem to be more related to dopaminergic or other transmitter dysfunction, meaning it's potentially reversible, that this can be modified, whereas more classical posterior cortical impairments, including memory and visuospatial deficits, are related to more posterior and more structural elements, including Lewy body pathology, but also Alzheimer's type pathology. So it's important to be aware of that. Nearly all Parkinson's patients, and at least most who have dementia, also will have some elements of Alzheimer's type pathology, particularly amyloid plaque, but also neurofibrillary tangles, and vice versa. When we see the brains of Alzheimer's patients, they will have some level of alpha-synuclein pathology. Lewy body pathology is very, very common, not necessarily at a large extent, but clearly present. So these diseases are well, there are different diagnoses and different diseases, but there's a lot of interaction, and you also know that there is a protein-protein interaction between tau, amyloid precursor protein, a beta, and alpha-synuclein. So if you have the changes in one protein, that leads to more frequent changes in the other protein metabolism. So I want to also talk briefly about another disease which is related to Parkinson's disease, which is called dementia of the Lewy bodies, which is roughly as frequent as Parkinson's disease. It's the second most common neurodegenerative dementia, and as you can see here, has a number of similar symptoms as Parkinson's disease. It's, in addition to dementia characterized by Parkinson's, the motor features of Parkinson's disease, but also visual hallucinations, specific parasomnia, REM sleep behavior disorder, and fluctuating cognition. And in that perspective, quite similar to Parkinson's disease and dementia. So we see these two disorders as part of a spectrum of Lewy body disease, and they are distinguished clinically by the fact that in Parkinson's disease you have a onset of motor symptoms and then dementia develops, while in dementia with Lewy bodies it usually starts with dementia or dementia and Parkinsonism at the same time, and then they progress. So that clearly shows, with this arbitrary 1-year rule, that these are diseases on the same spectrum. So it's important to also be aware of that in the dementia spectrum of diseases, we also have Lewy body diseases. So the pathologies or the mechanisms relevant for Parkinson's disease are also extremely relevant for other dementia disorders. Just briefly to show you this longitudinal study from Amsterdam, that although we have a progression from, at least on a group level, from normal cognition to early cognitive impairment, MCI and dementia, there's wide variation. So some patients move from worsening and then improvement, as I mentioned, but then tend to progress. So the progression is by far not linear, and one reason explaining that is that there are a number of factors influencing the cognitive functions of patients. It's not only the brain pathology, it's also associated symptoms, including depression. It could be various medications given to patients who can improve or worsen cognition. It can be insomnia or other sleep disturbances, which can also affect negatively the level of cognition, and of course other non-brain diseases which can affect the cognition. When we have patients with Parkinson's disease, many of them, as I mentioned, will be aware of that I have a disease with a high risk of dementia, and they will wonder, will I get dementia and when? And then we have both biomarkers but also clinical features which can guide us in terms of estimating that risk. And as you can see here, patients who have visual hallucinations, patients who have early mild cognitive impairment, who have hyposmia, who have this specific characteristic REM sleep behavior disorder, who have tendency to delirium, who have autonomic dysfunction. These patients have a higher risk of developing dementia compared to Parkinson's patients without these features. So to some extent, we can predict what's going to happen, but only to a certain extent. So we need biomarkers, and during the last years, there has been a number of biomarkers that have been shown ability to predict the risk for developing dementia. And we can see a number of imaging techniques, including MRI and FDG PET, but also more recently amyloid and tau PET, cerebrospinal fluid measurements, in particular the amyloid beta 42, which can peptide, which can be analyzed, and which is a marker of Alzheimer's disease and also a marker of cognitive progression, and EEG also demonstrating more and more a good capacity to predict dementia with a very quite specific Lewy body disease type pattern of changes on EEG. So this is one of a couple of early work demonstrating this fact that if you have Alzheimer type brain changes in the cerebrospinal fluid, that is then a predictor of subsequent cognitive decline and dementia risk. And unsurprisingly, because this is then a marker of that, in addition to the Lewy body pathology leading to Parkinson's disease, some of these patients will also have Alzheimer type pathology, which can be measured in the cerebrospinal fluid and other more recent markers. And then with this dual pathology, there is a higher risk of more severe disease progression, including dementia. So what about alpha-synuclein, the main culprit of Parkinson's disease and DLB? For many, many years, there's been very little progress. Yes, we can see some group levels in terms of cerebrospinal fluid alpha-synuclein levels, but nothing that can be used for individual patient prediction or diagnosis. However, during the last couple of years, there's been a tremendous development of seeding-based tests called RT-Quick taking from other prion diseases, the same techniques there, and then measuring the ability of patient-derived cerebrospinal fluid and the alpha-synuclein, which has a specific capacity to aggregate, and that can be done on a recombinant alpha-synuclein, and we can see that patients with disease tend to aggregate, whereas people without disease do not aggregate. So rather than looking at concentration, we look at now at the quality of the alpha-synuclein in the CSF, and also other biofluids can be measured in the brain mucosa, and there's also work ongoing in terms of blood. So those of you who have followed the news will have realized that there has been a fantastic progress in the development of blood-based biomarkers for Alzheimer's disease. And Nick Ashton, who was at King's and now in Gothenburg with Henrik Zetterberg and colleagues, has been leading that work. And we now have really good, based on new, more sensitive techniques and targeting in on the right markers, we now have very good blood-based measurements of Alzheimer's disease, which has now been implemented in the large clinical commercial trials of new Alzheimer's medications. So that's been a huge development just during the last couple of years. In terms of blood-based markers for non-Alzheimer's dementia or non-Alzheimer's neurodegenerative diseases, that's still more open, but there's a lot of promising work ongoing. And we can see here a list of markers which have been shown to to change in various diseases. We can see total tau and phosphorylated tau, but also synuclein measures and other relevant proteins which seem to have a signal in blood. Not yet at the stage where we can use them for diagnostic measure, but we can use them for prognosis. And this is one work for recently published work in DLB again, showing that patients with dementia with Lewy bodies have increased the levels of plasma p-tau181 and 231, the most promising markers. Less increase than in Alzheimer's disease, but increase compared to controls. And more importantly, when we have this increased level of these markers, those patients have a higher, more rapid cognitive decline. So we are getting blood-based prognostic biomarkers, which is a huge achievement and very helpful, and also helpful for clinical trials. And we can see here there's an apparent difference between DLB and Parkinson's disease, at least at early stage Parkinson's disease. These markers did not predict cognitive decline in a significant way. On the other hand, we had another marker, neurofilament light, which is a more global, non-specific marker of neurodegeneration, has been shown in our preliminary data, working with Ray Chowdhury and others in Parkinson Center here at King's, to be a predictor of more rapid cognitive decline. So we are getting close to having blood-based biomarkers of cognitive decline also in Parkinson's disease, which is has huge clinical importance. Just a few words about EEG. It's a biomarker which is not much used for a variety of reasons. We haven't yet identified really patterns changing in Alzheimer's disease that are really helpful, although using machine learning and AI analytic method, that work is progressing. But as mentioned earlier, Lewy body diseases, both DLB and Parkinson's disease, have specific features with slowing with more dominant slowing in the rhythms, and also a more variation in the brain rhythm on the EEG. Specific quantitative analyses have been able to separate groups with DLB from Alzheimer's disease and Parkinson patients with and without dementia, and also been able to predict which Parkinson patient will develop dementia based on their EEG pattern. Standard EEG, but more sophisticated quantitative measurement of the signal. So that's also a highly active area, and obviously a very tempting biomarker. It's non-invasive, it's cheap, and it's available everywhere. So it can be immediately implemented when we have sufficiently robust data. So then I wanted to sneak in a little bit of metal. So there are MRI is being used in Parkinson's disease, but not to a large extent. We can see more atrophy in relevant areas in people with Parkinson's disease and dementia. But in general, diagnosing Parkinson's disease, MRI is not the thing. We have dopamine transporters, nuclear medicine scans, but not MRI. However, more recent developments have identified MRI protocols, techniques that are sensitive to iron, and one of those are shown here in this review, and we can see, if possible, it's not easy to see, but we can see in the multisystem atrophy and other alpha-synucleinopathy, and also in progressive supranuclear palsy, that there are areas in the striatum which have an upload of iron, and there are also other techniques showing similar changes in people with Parkinson's disease. So we have metal-based diagnostic markers coming up. And another iron-based diagnostic is the use of transcranial sonography, where there is interesting data. Is not yet widely used, but particularly the German groups and Italian groups have worked on this, and we can see here that there is a pattern of higher echogenicity based on iron, probably not only iron, but mainly iron, increased iron levels in substantia nigra areas, which can be detected on sonography and ultrasound measurement. Obviously, quite sensitive measurement needs quite a number of training sessions in order to be able to do this, but it's interesting to have this opportunity, again, a non-invasive, cheap technique, and again, demonstrating the relevance of iron as a potential way of providing diagnosis. So then a little bit back to the clinic, and rounding up with some discussing medications. Just very briefly, it's important to know that we have medications which we now have some efficacy. We're far from a cure, but and no disease-modifying agents. But we have cholinesterase and glutamate-based therapies, which in meta-analysis have shown to have a positive effect, small but significant effect. Whereas for the others, it's much harder. The hallucinations in Parkinson's disease, etc. There are still challenges. So more work is needed, and whether working with groups like you and other specialists, and kind of using new models, whether that can help developing new treatment targets, that remains to be seen. But clearly, that's what we want to do, and that's how Giovanni and Joshua and myself are working towards. So here the meta-analysis evidence, don't need to go through that, supporting this. There are a number of non-pharmacological strategies that can be applied: cognitive training, physical exercise. We now have positive effects on the brain, and a variety of neurostimulation techniques, which are getting more and more, gaining more and more interest in, but still on the research level, ways of stimulating the brain function via magnetic or electric stimulations. A number of trials ongoing using different strategies. I want to mention this Entrin, the company Entrin has a gut-based agent, and there is quite robust evidence showing that, at least in many patients, Parkinson's disease and DLB patients tend to start that disease not in the brain but in the gut, and then addressing that as a therapeutic strategy is one very fascinating area. And we have nicotinic agonists studies ongoing, and Ambrexol, which is addressing the glucocerebrosidase and the GBA mutant patients. That's one interesting and important area. We have also P38 MIP kinase alpha inhibitor, neflamapimod, showing interesting findings. So there's a lot of work ongoing trying to identify neuroprotective agents which can protect the development of cognitive decline in Parkinson's disease. And just want to mention that we work a lot with many centers across Europe and across the world. And we have a multi-center UK studies also ongoing. So we are in a good position to test out new methods, diagnostic or therapeutic methods using our network of collaborators. And the absolute final slide, Richard told me lithium is a metal. So lithium, obviously, from a psychiatric point of view, very interesting and important drug. It's a fantastic treatment as a mood stabilizer. It's saved many lives in people with bipolar disorder. In addition, lithium has been known for many years to have a number of other potentially neuroprotective effects in affecting different pathways, including the GSK pathway, NF, and trophic factors, inflammation, etc. And that has been tested in several trials, whether we can use this substance to treat or prevent dementia. And there are interesting studies working on that, different doses, different strategies. I want to end with this. Large Danish registry-based studies, nearly 1 million of people involved, based on registries, where they looked at the incidence of dementia and compared that to the level of lithium in the drinking water. And as you can see here, the incidence of dementia is lower in areas with more lithium in the drinking water. So that's just a very fascinating way of applying Metallomics and preventing dementia. And with that, I end, and thank you for your attention.
Okay. Thank you very much, Dag.
Thank you, Dag, for a wonderful presentation. I'm stepping in for my colleague Wolfgang Marit, who can't chair this part, but we do have some questions in the Q&A, and I invite the audience to also add your questions to the Q&A for Dag. So the first question is actually from Giovanni. Is there clinical evidence that dietary compounds are able to cross the blood-brain barrier to improve deficits in cognition in patients? And perhaps some of these dietary compounds could be micronutrients or metals.
Yes, so thank you. So the answer to the first part is yes. Whether they are linked to metal, I don't know, but there is a lot of work showing association between the diet and the cognitive decline. So it has been more difficult to do trials, to randomize people to this or that nutrition, but there are also some data indicating that this is possible. So clearly, to the question of whether these nutritions cross the blood-brain barrier, that's a clear yes, which is a good starting point. And there are a number of very interesting supplements or dietary ingredients that need to be tested more thoroughly in that regard.
Thank you, Dag.
Thank you.
We have another question from Thea, who is wondering whether a large variability in Parkinson's disease presentations is observed in animal models. Of course, we're comparing animals and humans, but it would be interesting to see if there are changes in specific metal distributions in addition to iron that relate to certain presentations. Could this link to the use of PET metallomics, which we're going to hear about from our next speaker?
Yeah, thank you, Thea. That's a very good point. Our Parkinson animal models are not fantastic. So we have the classical, let's say, toxic lesion models, which are not particularly good on the cognitive aspect. We have genetic models, GBA, for example, also alpha-synuclein based, which to a better extent represent also the non-motor aspects of the disease. So I think, back to your question, I think that's definitely possible to using the animal models to see whether the Parkinson pathologies, how they are related to changes in metals. So yes, that's a good point, and it'd be great to look into that.
Great, thank you, Dag. If there are no more questions, we are just on time to move on to our next speaker. So with that, I'd like to again thank my colleague Dag Arslan for a wonderful talk. Please do continue to ask questions through the Q&A. And also for those of you who have just joined us, the full schedule and program PDF are available from the link which I have posted advertising this event. So our next speaker, I'm delighted, is Phil Blower, Professor Phil Blower, who is Head of Imaging, Chemistry and Biology in our School of Biomedical Engineering and Imaging Sciences. So we've had the privilege to work together with Phil in some of his PET metallomic compounds. And let me invite him now to tell you about his research. And of course, as my colleague Wolfgang mentioned, Phil was also instrumental from the outset in establishing the London Metallomics facility. So thank you very much for joining us, Phil, and I'll hand over to you.
Thank you, Richard. Thank you, Gio, for organising and inviting me again. Apologies to everybody, if anybody is here who was here last year and saw me. There are some minor differences between last year's talk and this year's talk. Let me just try and share the screen. Am I now sharing?
Perfect. We can see that, Phil. Thank you.
So, yeah, apologies again. Apologies also that I sort of the mirror image of Dag, the previous speaker, that I know about as much as about the brain as the gentleman in the image on the screen. But I do know about PET and trace metals, so maybe this meeting is serving a great purpose in bringing people who know about brain and people who know about metals together. So there's a list of acknowledgements which I want to get in first, because I do have a habit of running out of time and not acknowledging people properly. So I will try and acknowledge as many of those as possible during the talk. But there's lots of talented people on the left who've contributed, including the LMF team, and lots of funding agencies on the right. Very grateful to all. And I'm going to start with a slide which is not about the brain. It's really a plug for the LMF, because it's a lovely piece of work that George Firth, my PhD student who's now graduated, did in collaboration with Guy Rutter at Imperial and the LMF team. And this is a set of sections of pancreas in a knockout of the zinc T8 transporter, which is exclusively located in the islets of the pancreas. And you can see that in the wild type images on the left, there's a very at the top you've got an image from using laser ablation of zinc distribution, and the zinc distribution is very high in the islets. If you go across to the right, you see that in the homozygote knockout zinc T8 mouse, there is no accumulation of zinc in the islets, and you can see that the islets are there because of the deficiencies of other elements that are associated with the islets. So you can identify that you are looking at islet tissue, but zinc is absent. So you can see with conventional, well, the modern Metallomics laser ablation techniques, that there is a very marked non-equilibrium distribution of trace metals within tissues at the cellular level. So these data, the data that you've seen and the data from the kind of techniques that Alex and Thea introduced earlier on show you how much metal is present and where it is at the cellular level, and it can do it for lots of metals at once. So it's very powerful and very comprehensive. But there's a big but, and that is that the mouse is dead. it is a very invasive method. It requires tissue samples and tissue sections, sometimes biopsies, but usually autopsy. And so it's a snapshot at a single time of the distribution of the metal that has resulted from a long period of equilibration and distribution. What would complement this would be to measure the dynamic fluxes of metals. How is that snapshot state reached? What are the acute processes that lead to it? We'd also like it to be non-invasive so that we can apply it to humans, living humans, either healthy humans or diseased humans, and use it in not only for research, but in their diagnosis and monitoring of their therapy. And of course, it has to be non-invasive and harmless to do that. And one of our solutions to that is using radionuclide imaging. And this is we've kind of rather cheekily coined the term PET metallomics. To get the kinds of images that you see on the screen now, the left one actually is not a metallomic image, but it is a PET image. It's an image of the distribution of the sodium iodide symporter using a radioactive substrate of the sodium iodide symporter, F-fluorine-18 labeled tetrafluoroborate. The one on the right, those of you who are more biologically inclined will realize that that's a mouse. The one on the right is a human, and that is a metallomic image because it's the distribution of copper that is injected as ionic copper-64, a positron emitter, either intravenously at the top or orally at the bottom. And I've put it there, I'll show it again later on. It's just there to show the kind of image that you get. You can take a picture of where the radionuclide is and hence where the metal is at any time within a reasonable multiple of the half-life of the radionuclide. And you can track this non-invasively with a scanner. And this is the field of nuclear medicine, which is a common everyday diagnostic technique in nearly all hospitals, where we inject a radioactive compound or drug into a person, and we do this on purpose for the benefit of the patient. The compound is carried around the body in the blood. Some of it binds to specific targets in the body, via molecular recognition or some kind of metabolic process, while the rest is excreted. And we map the location of the radioactivity across the whole body of the patient with a scanner, either a gamma camera or a PET scanner, positron emission tomography. And the versatility of the approach and its diagnostic value come from designing the radioactive molecule, and it may just be a metal ion, to bind to a particular molecule or receptor or molecular process in the body, and hence give you an image of where that process is happening. We use radioactivity because of the tissue penetration of the rays. So whether the radiation comes from a gamma emitter, so it's gamma rays, or whether the gamma rays originate from the annihilation of a positron with an electron, it's penetrating. It's easily detected outside the body. There's very little attenuation. It's also extremely sensitive in terms of the signal that you get from a very tiny amount of the compound. So the radio tracer in the body is at picomolar levels. So this is really a truly tracer concept. You can do this and measure things without really perturbing the system that you're investigating. In contrast to the use of MRI contrast agents, where the amount of the contrast agent is much higher, typically millimolar, but at least 1 million times higher than a PET tracer would be. So we're looking at imaging molecular processes. It's known as molecular imaging. On the downside, of course, the patient will get a radiation dose. There may be some toxicity associated with that, but that is kept in tolerable limits because we know a lot about the effects of radiation. And in particular, with the new technology that is emerging and probably will be established in the UK in the next 2 to 3 years, total body PET, which images the whole body at the same time, instead of scanning a patient linearly through the scanner, that will reduce by at least 10-fold, maybe 40-fold, the radiation doses to the patient. So much more research-based and less medical-based scanning will become possible. It's also expensive. The radionuclides can be expensive and the technology to do the scanning is expensive. And another thing about radionuclides is they're not smart. The only smart content is that they are attached to a molecule of your choice, but the signal itself cannot be switched on and off. A radioactive decay just happens when it's going to happen and you have no control over it. So it doesn't reflect the environment in any sense, except that the concentration of the particular receptor or the molecular cause of the accumulation is visualized. So we have radionuclides that can be imaged in this way from across the periodic table, and I'm putting this in just as a sort of a show of the versatility, of the kinds of the range of elements that we can use. All of the ones that are flashing up are routinely used in nuclear medicine, either for therapy or for imaging. And we hope over the next few years to add some more to make it feasible to really investigate metallomics. Now, metals have been used, radioactive metals have been used in nuclear medicine ever since nuclear medicine started, but the way they're normally used is as a radiolabel for a biological molecule. So a molecule like a peptide in this case on the screen, this is a somatostatin analog, and appended to it is a chelating agent which has a radioactive gallium attached. And the gallium is there just to provide the signal. It doesn't do the targeting, it's just along for the ride. So it's the targeting biomolecule, the somatostatin analog, that is telling you about the biology going on in the patient, imaging somatostatin receptors. What we're doing is moving away from this and using the metallic radionuclides to investigate the biology of those metals, so PET as a servant of metallomics. And we're developing an expanding array of positron-emitting isotopes of biologically essential metals, or even some toxic metals where we need to investigate their biology. So a range of copper isotopes are established, and I'll show you some examples. Isotopes of manganese are beginning to emerge. There's very little being done, but I'll show you a little bit of at the very beginning of this field. Zinc isotopes are just about emerging and are slightly ahead of manganese, but a long way behind copper. And some of the older work on cobalt 55, iron 52 is out there in the literature from many years ago, before PET really came along. And as well as isotopes of biologically essential metals, in the cases where there are no useful isotopes of those metals, we can learn something by looking at the biology of surrogates using radiolabeled, for example, rubidium as a surrogate of potassium, or thallium as a surrogate of potassium, and gallium as a surrogate of iron, and so on. We have to be very careful, of course, not to overinterpret those, but we can learn something. So an example of using rubidium as a potassium analog is here on this slide. We have this is used for myocardial perfusion imaging, because rubidium-82 is a positron emitter with a half-life of 75 seconds, and it is a substrate for the sodium-potassium ATPase, just like potassium. And the heart, of course, has an awful lot of sodium-potassium ATPase. It's pumping like crazy. So there is a lot of rubidium uptake in the myocardium, more or less proportional to the perfusion. So we can use it to do PET imaging of myocardial perfusion, as in the image on the top right. Recently, there's been some interest in pushing that into the field of oncology as well, because sodium potassium ATPase chain is often increased in tumours. So we can measure tumour perfusion, but we can also at least work towards extracting from those images some measure of the function of the sodium potassium ATPase. Gallium as a substrate of iron is possible because of its chemistry, not necessarily directly related to its role in the body, but it's just its similarity to iron in some cases. So we have two isotopes, gallium-67, which is a gamma emitter with a half-life of about 3 days, and gallium-68 with a half-life of about 68 minutes. When you inject ionic gallium or weakly chelated gallium into the bloodstream, it binds to transferrin immediately. So what you tend to be imaging is a distribution of the transferrin receptor, and that's what the image on the right shows. This is a very old gamma camera image of a patient with lymphoma, and the lymphoma is there present at the base of the neck, and the lymphoma is associated with high level expression of transferrin receptors, so we can highlight the tumor on the image. Another application of this is exploiting the similar structure when a siderophore that is produced by microorganisms to bind iron binds gallium in a very similar way and produces an isostructural complex. And that complex is recognized by microorganisms in their quest to accumulate iron from the environment. And so making gallium-68 labeled siderophores may be potentially useful for imaging infection. And we at the moment at King's have a trial of gallium-68 desferrioxamine B, which is a gallium siderophore complex, looking at detecting vascular stent infection in patients who've had a stent implant and at risk of infection. So molecules of like this. Ionic copper is perhaps one of the most well developed in the field of metallomics. This is an example where copper can be used, just copper chloride or copper acetate can be injected intravenously, and it shows up prostate cancer because of the increased expression of CTL1, the main copper transporter. On the left is a mouse with an implanted copper accumulating tumor, a prostate cancer, and on the right is a human. On the right-hand image is a PET scan. It's a transverse section through the base of the pelvis, and you can see a couple of prostate cancer lesions which are visible because they are taking up copper like crazy. And in prostate cancer, the malignancy the high zinc uptake in the healthy prostate goes down, and there is an increase in copper uptake. And this is very useful because the copper is not present in the bladder, so you've got a very clear field of view around the base of the pelvis to detect prostate cancer. So the message there is that copper-64 imaging, I'll go forward again, can be used in tumor imaging in humans, and probably what we're seeing is increased expression of the CTR1 copper transporter. What about a disease that is really about copper? Wilson's disease. It's a genetic disorder of copper transport caused by mutation or loss of the ATP7B copper transporter. And diagnosis in Wilson's disease is difficult. Liver biopsy is one of the standard methods of diagnosis, very invasive procedure. Copper chelation therapies are available to relieve copper overload, but their use is not well understood. It's a bit unpredictable, can have side effects, and its effect on the neurological symptoms of Wilson's disease is unpredictable. Sometimes it seems to make them worse. So clinicians would like to have a non-invasive test to quantify copper accumulation in and excretion from all the organs, not just one, but across the body, particularly the brain, because the brain can be badly affected and neurological symptoms occur, and that can be associated with chelator administration. So we want to be able to predict whether chelator therapy will affect neurological symptoms positively or negatively. And a study of this in animals was now about 10 or 11 years ago, came from University of Texas, where they have a knockout, ATP7B knockout, which certainly does affect the uptake of injected radioactive copper in the liver. So these are PET scans of small animals on the left, and they can be quantified. So on the right, inside the red box, you can see that the liver uptake in particular of radioactive copper over the first 60 minutes in the wild type is pretty flat. It's very quickly accumulated, and then it reaches a balance with copper flowing out of the liver around to the rest of the body, and copper being accumulated into the liver from the blood. But in the knockout mouse, the excretion of copper from the liver into the gut and to the rest of the body slows down, and you end up with a steadily increasing uptake of copper in the liver. So you can see the effect of that genetic abnormality by PET scanning. And in this slide, this is much more recent. Just last year, it was shown that you can very much do this in humans as well. So on the left is a healthy human who's been intravenously injected with radioactive copper chloride in the top, showing high accumulation in the liver and gradual excretion from the liver to the gut and a little bit of shipment to the rest of the body. The bottom is oral administration, and you can see that a lot of the copper is staying in the gut, and only a fraction of it is being absorbed into the liver. But apart from that, it's very similar. But if you look at a population that includes a cohort of patients that includes patients with Wilson's disease compared to normal controls, you can see very much the same thing happening as happened in the ATP7B knockout mice. In the controls, the copper in the liver stays relatively flat over the first few hours, but in the Wilson's patients, it is continuing to increase over those first few hours. So you have a diagnostic method of using PET with copper-64 to accurately diagnose Wilson's patients. And so this is still in the very early days, but it clearly shows that it warrants further clinical evaluation. What about the brain in those Wilson's patients, or indeed any patients who have are at risk of dementia or neurological symptoms? If you inject ionic copper, actually very little of it gets to the brain, so you cannot do very much in the way of meaningful imaging in the brain. But we do have ways of bypassing that problem. So the compound shown in the middle of the screen is copper GTSM. It's a lipophilic planar compound that crosses the blood-brain barrier and gets into cells, but when it's exposed to the intracellular environment, it's very easily reduced to copper 1, and the copper 1 is released into the cell and is essentially trapped there. So the PET images at the bottom, the two on the left, show what happens in a mouse if you inject just ionic copper acetate. And on the right, if you inject copper GTSM, you see uptake in all tissues, particularly those that have a very high blood flow, and you can see the heart lighting up. But you can also see the contrast in the brain between copper GTSM and copper acetate. There's almost nothing in the brain after copper acetate injection, but the brain after copper GTSM injection retains a lot of radioactivity, and you can see the brain and the spinal cord really clearly. And that is shown qualitatively on the graph on the right. The bar chart shows that the GTSM has a very high brain uptake compared to the copper acetate. Interestingly, most tissues over the next 24 hours clear the copper that they have accumulated to a large degree, but oddly enough, the brain doesn't. The brain uptake, if you look at the graph in the top right-hand corner, the light blue and the orange lines are essentially horizontal at the bottom there, whereas the other tissues are clearing their copper. The brain doesn't seem to have a way of getting rid of that copper very quickly, which I think might be an interesting observation to take forward into the whole field of brain metallomics. So can we use this to study the distribution of copper in the brain? Well, the answer is yes. We don't quite know what it means yet, but both our group and Paul Donnelly's group in Melbourne have used this approach in two different Alzheimer's mouse models. Both gave very similar results. Copper GTSM, copper acetate or copper chloride showed very little brain penetration, but the bis-semicarbazone copper, the copper GTSM, does enter the brain, and it enters the brain about 40% more in the case of the mutant animals, the Alzheimer's models, than it does in the healthy mice matched to the same age. And it also clears a bit quicker from the disease animals compared to the normal animals, but it's still very, very slow clearance from the brain. Another mouse model of dementia, which we also investigated, a Niemann-Pick-C model, also showed very similar behavior. So we asked whether this can be used in Wilson's disease. Nobody has done this yet, but we do plan to try and do this over the next couple of years. And the Wilson's disease community is very interested in getting involved with that. Menkes disease is another disease of copper abnormality of copper transport, this time resulting in deficiency of copper in most tissues. And this was also investigated by using PET in animal models. And this is an ATP7A mutant. And you can see on the right where a wild type mouse and a Menkes disease model were injected with radioactive, just copper chloride. And you see a different biodistribution in the two cases. So you can see in the brain, for example, on the far right, the graph shows that the red line shows less uptake of copper in the brain of the Menkes model compared to the normal animals. What this group also did was to investigate the effect of chelation therapy on the distribution of copper. And in these experiments, they gave the animal a dose of either disulfiram or penicillamine, either of which are chelation therapy agents, to investigate what the effect of a subsequent injection of radioactive copper is. And you can see in the case of the penicillamine, which is the yellow line in the graph on the bottom right, there is less, it results in less uptake in brain, but the disulfiram results in a massive increase in the injected copper in the brain. So it looks as if The disulfiram is doing the same job as the GTSM was in the previous experiment. It's binding the injected copper in blood. This is a bit of a hypothesis, but this will explain what's happening, and delivering it in lipophilic form across the blood-brain barrier and releasing it in the form of copper after a bioreductive process. So it is clear from this experiment that chelation therapy can really drastically interfere with the distribution of copper and possibly other metals and its delivery to brain. And PET imaging offers us a way to investigate this. We've done some experiments with a different chelation therapy. This is tetrathiamolybdate, a chelating agent for copper that's under a clinical investigation at the moment. We studied the distribution of copper GTSM and copper chloride as affected by tetrathiamolybdate. We did the experiment slightly differently. We injected copper and waited for half an hour for the copper to reach its equilibrium distribution, and then injected the tetrathiamolybdate. So there's lots of ways of doing these experiments, and we need to do them in all the different ways in order to learn what we can. But on the next slide, we can see that there was an effect, and it was a very odd effect. The copper that was deposited in the liver and spleen and the heart was investigated with a subsequent injection of tetrathiamolybdate. And you can see that in the middle graph, the activity in the heart was cleared more slowly from the heart muscle in the animals that were given tetrathiamolybdate, which was quite the opposite of what we were expecting. Similar experiments on the brain didn't show very much change, but these are very preliminary experiments, and they show what you can do with PET to investigate the pharmacological activity of chelation therapy. So the message from those experiments is that we can image effects of defective copper transport genes for clinical diagnosis. Clearance of copper from brain is very slow, and this may be an important observation in meta-related brain pathology. And we can image the effects of chelation therapy, and we see even at the beginning of those experiments that redistribution to brain can occur. Zinc is another one, and I'm going to go. Zinc has been just touched upon in the last 2 or 3 minutes. I just want to show examples of using zinc 63, a positron emitter with a half-life of 38 minutes, and this was done by a team at the Mayo Clinic, who looked for the very first time at zinc 63 in humans, and they chose to compare healthy elderly patients with Alzheimer's patients, and they showed differences in the brain uptake and clearance of ionic zinc between the Alzheimer's population and the healthy population. That study was very limited by the very short half-life. You could only image for a couple of hours. So we've been looking into an alternative radionuclide of zinc 62, which has a half-life of 9 hours. To cut a long story short, George Firth, my PhD student, has shown that this zinc 62 can be used to image zinc distribution. And I'm going to skip this slide in the interest of time because it's rather a technical slide. But what it shows is zinc is taken up in the pancreas. And I'm going to refer back to the original slide with that zinc T8 knockout mouse, where the PET study shows some complementary results compared to the laser ablation study. That same mutation, or that same knockout that gives rise to a failure to take zinc into the islets, does not affect the uptake of zinc in the pancreas. The uptake of zinc in the homozygous mutant mice and in the wild-type mice was exactly the same. It was very high in all of them, which implies that there are a different set of transporters involved. So this is an example of how the PET technique can be complementary to the microscopic static and snapshot distribution that you get from laser ablation. Just to finish off, I want to mention that we're just beginning to do studies with manganese. We have done very little except to look at how manganese behaves in normal mice. Like zinc, it's taken up in the pancreas, taken up in the heart initially, but it clears from the heart over a period of hours. And we anticipate that this could be used in studying manganism and also environmental toxicology. And one of the uses that manganese has been put to in human or in animals is to image neural connectivity by injecting manganese on a large scale so that it can be detected by MRI. and watching the manganese diffuse along neural tracts. So you can determine whether there's any, what the neural tract looks like, and you can determine whether, for example, nerve repair has taken place, and you can determine whether traces can reach the brain and travel along pathways, axon pathways after administration into the nose, for example. And in the case of mice, it certainly does. You can see that radioactivity moves from the nasal cavity along the olfactory tract into the olfactory bulb and beyond. So we can use PET imaging in preference to MRI, because PET imaging uses a much lower amount of manganese, undetectable in chemical terms, whereas manganese studied by MRI has to be present in quantities that are actually too toxic to do this routinely. Finally, we have been applying the same techniques to find out whether environmental particulate pollutants can penetrate the brain if inhaled. And these are just very preliminary experiments in mice, where nanoparticles labeled with radioactivity are administered into the nasal cavity, and you can watch, using PET imaging, whether those particles are translocated into the brain. And in these mice, there's no doubt that they are. So we do have something to worry about. And if we apply this to humans, when we've developed ways of simulating environmental particulate pollutants, radiolabeled with potential pollutants like manganese, we can trace not only whether those particles penetrate the brains of humans, but also what happens to the manganese after it does get into the brain. So just to summarize then, laser ablation and other elemental imaging methods can produce cell level distribution of the element content, and it's a static snapshot after a prolonged accumulation, and it's invasive. Very limited applications in humans. Radionuclide imaging methods provide us with a short-term, over a period of minutes or days or possibly weeks if we go to total body PET, measurements of trafficking at the whole body and organ level, and it's dynamic and short-term, and it's non-invasive, and it's repeatable, and we can do it in humans. So the methods are complementary. So to finish, we have the tools to do PET metallomics now, or very soon we'll have a few more of them in animal models and in humans, and we look to this community to bring us questions to answer, because like I said, I don't know my thalamus from the elbow, I just know how to do PET. Thank you very much for your attention, everybody.
Thank you so much, Phil. Wonderful talk, and you're leading into the next type of talks about manganese. I have a request from the organizers here that any questions are put to you by email, because we do not have much time. Is that fine with you, Phil?
Yes, absolutely, it's fine with me. Email is the best way, because I am in the middle of another symposium at the same time, like some of the others. So
and I apologize for that too, not being able to introduce.
Yeah, do email me. We are actively looking for applications, because we're in the business of developing tools, and we're not so aware of the questions that we would be able to answer using those tools. So please do come. And like the London Metallomics facility, we are also in the business of creating a hub which people can come and use. So, you know, we're anxious to set up collaborations and entertain visiting researchers to come and do studies of this kind.
Absolutely. Thank you, Phil. So I will hand it over to my colleague, my longtime colleague, Professor Krista Hawkestrand, who will introduce the next two speakers. It's up to you, Krista.
Thank you very much, Wolfgang. So our next speaker is going to be Dr. Karen Tushel, and she is working as a consultant metabolic pediatrician at the Great Ormond Street Hospital here in London. And she is a MRC clinical scientist fellow at the UCL Goss Institute of Child Health, and she will talk to us about inherited manganese transporter defects in the brain. And over to you, Karen.
Yeah, hello everyone. I hope you can hear me and see my screen as well. Can you just confirm that?
Hear you.
Yeah, okay, perfect.
See you. Have the screen.
Perfect. All right. Thank you. So thank you very much for the invitation today for me to present my work. It's a real pleasure to share my work with the Metallomics community. And I will give you a little summary of our work that sort of tries to span basic science and translational research. And mainly our work focuses on inherited manganese transporter defects, particularly those that lead to manganese overload and neurotoxicity. And we're using zebrafish models to study the mechanisms of manganese neurotoxicity. And we have an additional translational aspect and hope to develop sort of more effective oral manganese chelator. I'm sure people here are familiar with manganese and know that manganese is an essential trace metal that's required for normal brain function, because it's a constituent of metalloenzymes and also used as an enzyme activator. And any disturbance in manganese homeostasis leads to neurological disease. That holds true for both manganese deficiency and manganese overload. And as said, my group focuses on disorders related to manganese overload. And when we have too much manganese on board, it accumulates in the basal ganglia, particularly here in the globus pallidus in the brain, and causes a movement disorder, a Parkinsonian type movement disorder that is associated with psychiatric and cognitive defects. And there are various ways of manganese overexposure, so acquired causes that include occupational overexposure in mining and welding industries, for example, but also environmental overexposure through contaminated drinking water or air pollution. And there have been cases of accidental toxicity, for example, giving manganese chloride instead of magnesium chloride. Users who've been using potassium permanganate to synthesize the ephedrine have experienced manganese neurotoxicity. Equally, manganese is present in parenteral nutrition and can be a source of manganese toxicity. So we're mainly interested in the inherited types of manganese transporter defects that are caused by mutations in genes encoding manganese transporters, such as SLC30A10 and SLC39A14. But we also see manganese neurotoxicity in end-stage liver disease, where the liver simply can't facilitate the biliary manganese excretion. And we're aware nowadays that metal dyshomeostasis, as well as manganese dyshomeostasis, is a feature of Parkinson's disease and Alzheimer's, so common neurodegenerative disorders. And I want to take you, all the way to the beginning of my work. So this is the first patient that we looked after at Great Ormond Street with an inherited manganese transporter defect, and she presented with a Parkinsonian movement disorder. So you can see here that she's got a reduced arm swing, she walks quite slowly and has a characteristic high stepping gait. She also has got dystonia in her upper and lower limbs, which is like an increased muscle tone that makes it difficult for her to perform sort of rapid fine motor movements. She also had liver cirrhosis and she had significantly raised blood manganese levels. She had polycythemia, which is an increased red blood cell count, and she was iron deficient. You can see here her MRI, so you might be able to appreciate again this typical appearance of manganese on the T1 weighted imaging. So manganese is a paramagnetic metal and thereby reduces the relaxation time on the T1 imaging. You can also see that here in the brainstem, the dorsal pons is very hyperintense. And then on the TT2 weighted imaging, you get hypointensities in the corresponding areas in the globus pallidus. When we look at her liver MRI, you see that her liver MRI compared to the control is very bright. So again, hyperintensity caused by accumulation of manganese. And the liver biopsy confirmed the liver cirrhosis. At the time, we sort of accumulated other patients with similar phenotypes. And most of them were actually from consanguineous families. So as you can see, these were first cousin parents, which suggested that this is most likely an autosomal recessive disorder. So I at the time performed homozygosity mapping, and that suggested SLC30A10 as a candidate gene. At the time, SLC30A10 was thought to be a zinc transporter expressed at the plasma membrane to facilitate zinc export. It was known to be expressed in liver, intestine, and brain. And I then went on to perform some functional studies in yeast. So these are overexpression studies with the wild types construct and then constructs containing the patient mutations. And while you can see that the wild type construct can rescue a manganese sensitive yeast strain, the patient mutations can't. So there we've sort of shown that SLC30A10 indeed facilitates manganese transport. Subsequently, I also generated a zebrafish loss of function mutant using talents at the time. And you can see that from 2 weeks post-fertilization, these zebrafish start to accumulate more manganese compared to the wild type. And this is our current understanding of the disease pathogenesis. So we take up manganese with the diet, and it's absorbed in the small intestine, enters the enterohepatic circulation, gets into the liver, and SLC30A10 is the transporter that we need for biliary manganese excretion. And whenever you have mutations in this protein, you get the overload of manganese in the liver, causing liver cirrhosis. It also leads to activation of the HIF pathway, which then leads to increased expression of erythropoietin, hence the polycythemia and increased red blood cell count. And the systemic manganese overload again, leads to the accumulation of manganese in the brain, particularly the basal ganglia. At the same time, we then had a few patients who seemed to sort of share the neurological phenotype. So they also had a sort of typical movement disorder and became wheelchair bound already quite early during childhood. However, they didn't have any liver cirrhosis, and they also didn't have the polycythemia. And most importantly, they didn't have any mutations in SLC30A10. So by that time, we had whole exome sequencing available. And sorry, I just first want to also show you the MRI brain. So the MRI brain, again, has got the typical appearances of manganese accumulation. And we had a post-mortem on one patient who unfortunately passed away. And you can see that in the globus pallidus, you do have neurodegeneration. I mean, there's one lonely neuron, but you can't really see any other neuronal cell bodies. And in the white matter, you have this big vacuoles. So you also have the affection of the white matter. When you look at the MRI of the liver, you can see that this is an affected patient that doesn't carry the SLC30A10 mutations, while this is one with SLC30A10 mutation. So you see that in these patients, we don't really have any manganese accumulation in the liver. And then whole exome sequencing identified SLC39A14 as a candidate gene. So it was known to be a divalent cation and influx transporter, and that it could transport zinc, iron, manganese, and cadmium, at least in vitro. And we then went on to measure metal levels in 2 patients with different mutations and one set of parents, but they had an isolated increase in manganese, while zinc, iron, and cadmium levels were normal. And then did some overexpression studies in HEC293 cells. So again, you can see that the wild type construct facilitates manganese uptake, while the ones, the mutants, sort of have reduced manganese uptake. And this is our current understanding of what we think is going on when this transporter is not functioning. So we believe that SLC39A14 is the transporter that we need in order to get manganese into the liver and also into the enterocytes for subsequent excretion through SLC30A10. So obviously when you have mutations in this protein, then you get the systemic manganese overload and the accumulation in the brain. But this also explains why we don't see liver involvement in this disorder. So then also here, I developed a zebrafish loss of function mutant using CRISPR. What's different in zebrafish is here you look at the expression of the transporter, and this is the pronephric duct, so the kidneys. So probably in zebrafish, rather than the liver, it is probably the kidney that is facilitating the regulation of manganese homeostasis. And this loss of function mutant shows obviously the reduction of the SLC39A14 transcript. And we see manganese accumulation from 5 days post-fertilization. and then obviously also a massive increase in manganese in adult zebrafish brain. When we expose the zebrafish larvae to increased concentrations of manganese chloride, you also see that they have increased sensitivity to manganese toxicity. So here the lethality on the y-axis, so they pretty much die earlier than wild-type larvae. When we look at the locomotor behavior, so here you see the wild types in blue and in red are the mutants. When you expose them to manganese, you see that the locomotion is very much reduced. And in a first attempt to sort of further study the mechanisms of manganese neurotoxicity, in a collaborative project with the Bush Lab, we did RNA sequencing on an incross of the SLC39A14 larvae, so heterozygous incross, and we exposed half of them to manganese and half of them were untreated, and then analyzed the changes in gene expression. And the sort of most interesting group of genes was definitely the one here that when you compare the manganese treated mutants with the manganese treated siblings, you can see that there were about 600 genes that were differentially expressed in the mutant, but not in the treated wild type, and obviously not in the treated wild types or mutants. And when we looked at the zebrafish anatomy terms for the differentially expressed genes, they all pretty much map to brain and eye, which is consistent with their neurological phenotype. And analyzing the gene ontology terms, they brought up terms related to, for example, the unfolded protein response, so suggesting ER stress. It highlighted genes in relation to phototransduction and also translation, RNA binding, and ribosomes. There was also a group of calcium-related genes that was differentially expressed. So we then, looked at the first step at calcium levels in the manganese-treated wild types and mutants, and you can see that in both wild types and mutants, treatment with manganese leads to a significant lower level, like whole body calcium level, as well as magnesium. Well, obviously your manganese treatment causes increase in your manganese level. In the next step, we were interested whether we do see apoptosis in manganese-treated mutants and wild types, but at least at this concentration and for the exposure time, so we always expose from like 2 or 3 days post-fertilization until 6 days, we don't really see any increase in apoptosis, which would suggest that the changes we see are most likely functional changes rather than neurodegeneration. And in order to sort of identify brain regions that we now want to study further to see what's actually going on in the brain, we then did a neuronal activity mapping using in situ hybridization. And what you can see here is the wild type and the mutant compared. And this is actually a comparison between the untreated and the treated wild type, and here the untreated and treated mutant. Anything that you see in green is suppressed brain activity, and anything in magenta is increased brain activity. And although I don't expect you to know what you're looking at, but this is actually a brain from above of the zebrafish larvae. And this area here, and there is the optic tectum. And you can see that in the optic tectum, that obviously processes your visual information, you've got reduced activity, more pronounced in the mutant. And additionally, in the mutant, in the telencephalon, you have changes with increased neuronal activity. And this obviously gives us now like the first sort of indication of where we will look for sort of changes in neurochemistry, etc. Then obviously, because of a big group of genes related to phototransduction that show changes in expression, we then looked at the vision phenotype of these larvae. And you can already by eye tell that the manganese-treated larvae, the mutant larvae, they're much more pigmented. And we know that when, for example, zebrafish are blind, they are darker because they don't contract the melanophores. And when we then look at the optokinetic response, you see that at the high spatial frequencies, the mutant larvae have a reduced eye velocity. then, look at the retinal histology. It doesn't suggest any structural changes. So again, making it more likely that manganese causes some functional changes. And Arthur is a postdoc in my lab. He's now looking at the retinal phenotype further, because obviously if the retina has got or most of the neuronal subtypes we also see in the brain. And if we start to understand what's going on in the retina, we'll probably also better understand what's going on in the brain. Obviously, I need to mention that here as well, in a collaborative project with the London Metallomics facility, we have started to look at metal distribution and with the view to also look at metal interactions, given that most of these transporters are not just manganese transporters, but they can also transport other metal ions. And Golzana in the lab, she's a LIDO PhD rotation student. So she started to do some sectioning, and we've been running some laser ablation ICP-MS experiments with Alex Griffiths. I must apologize for the poor quality of our sections in this run. They've improved much, and we had a run again last week, for which we're now waiting for results. But I think so what you see here is you're looking at the zebrafish larvae from above. So these are the eyes, and this is the laser ablation ICP-MS result. If you compare that to the treated mutant, I'm sure you can appreciate that there is more manganese accumulating around the eye. So these are really just very preliminary results, and we've now sectioned the whole brain from the front to back, and we're excited to get some results soon. What was also quite interesting when we looked at the RNA sequencing data of the SLC39A14 mutant, we had a group of genes that was differentially expressed in the untreated mutant compared to the untreated siblings. And once they were treated, these expression changes normalized. So this is really only possible if there's also a degree of manganese deficiency. And that is plausible given that SLC39A14 is a manganese uptake transporter. So in that case, it's possible that, for example, in some organelles or in specific tissues, you have additional manganese deficiency in parallel to toxicity. These differentially expressed genes, they pretty much only map to the brain. And when we look at the gene ontology terms, again, there were a lot of genes differentially expressed that are related to cell-to-cell adhesion. And these are probably all due to 10 different protocadherin genes that are differentially expressed. We also had some presynaptic genes differentially expressed that are required for neurotransmitter release. Again, many of these genes are related to calcium, and we had some other calcium-related genes differentially expressed that was different to the set of genes we saw in manganese toxicity. So we're pretty sure that calcium dyshomeostasis plays a role in both manganese toxicity and deficiency. Doesn't want to move. No. Okay, so in summary, loss of SLC39A14 causes manganese neurotoxicity and manganese deficiency in our zebrafish model. And the transcriptomic changes really affect solely brain and eye. And calcium plays a central role in manganese dyshomeostasis. And we hope that in the future we'll be able to now, determine the hierarchy of the downstream effects of manganese neurotoxicity. So next, I want to give you a summary of our slightly more translational work and our progress with a manganese chelator development. So I need to mention that in clinic, we're using the sodium calcium editate as chelation agent to treat manganese overload, which works quite well. I mean, you can see that if you compare the two MRIs pre and post treatment, we can definitely mobilize manganese from the brain, and clinically patients do improve. So again, showing that we definitely have a reversible component of manganese toxicity. But the problem with the sodium calcium editate is that we have to give it intravenously through quite burdensome five-day courses every month. Obviously, patients need to have long-term venous access that carries the infection risk. And if they have to go to hospital for 5 days every month, that obviously interferes with schooling and employment and is incredibly expensive. Many of these patients are at home in low-income countries such as India. And therefore, there are very few patients who are actually being treated with disodium calcium meditate. So our ideal manganese chelator should really be orally bioavailable, should cross membranes, and should be manganese specific. And in a collaborative project with John Spencer and George Kostakis from Sussex University, we found one manganese chelator. That structure I can't share yet with you because of IP reasons. But what I can show you is that we've been screening, obviously, our zebrafish mutant. We know that they have a locomotor phenotype. So what you can see here is the locomotor tracked over 3 days, from 4 days post-fertilization to 6 days post-fertilization. And the fish are in a 96 well plates, and their movements are continuously tracked. Here is when the light is on. This is during the night when the light is off. Again, light on, off, on, off. And when we look at 6 days post-fertilization, so in green here is the manganese treated mutant, and all the others, the untreated mutant, and the treated mutant with the chelator. Sort of you can see that it rescues that locomotion phenotype. When we analyze this here at 6 days post-fertilization, you can see there's a significant difference between the chelator treated and the untreated mutants. You can even by eye tell that the chelator has an effect, because what I said before to you, that the pigmentation phenotype is sort of reverted again with the chelator treated larvae. And when we analyze manganese levels with ICP-MS, you can see that the chelator has a massive effect. So it pretty much normalizes manganese levels in the chelator treated mutant. When we then compare this to the effect of disodium calcium meditate, so disodium calcium meditate doesn't have a massive effect on manganese levels when we just add it to the fish water. And we've done some log case studies as well to see what's the manganese affinity, and it is much higher with the new chelator compared to EDTA, but it also has an even higher affinity to zinc than it has to manganese, and that obviously could sort of cause problems in the, if you use it long term. Certainly when we just look at sort of 3 days treatment, it doesn't have any effect on zinc. And to be honest, even with EDTA in patients, we don't really see any problems with zinc when they're treated with disodium calcium meditate. Sometimes we see lower levels and then we just supplement zinc. So maybe the manganese specificity doesn't need to be 100%. And obviously we just add, so in the first screen, we just add the chelator to the manganese containing water. So we obviously wanted to make sure that we don't just see an effect of manganese chelation within the water. So we then injected the chelator into the heart, and we also looked at a methylated compound here, and you can see that by injection into the heart you also get an effect on manganese levels. Same again, zinc, iron, copper and calcium levels are unchanged. And this is just in comparison to disodium calcium meditate that we know sort of has an effect in patients, and the effect is pretty much the same for both compounds. So as future work, we will do further compound screening in zebrafish, because the sort of quite promising compound that we're working with at the moment, we may struggle to formulate that into an oral chelator because it's quite hydrophilic. And ultimately, we want to look at the best candidate in the SLC3010 knockout mouse, and I'm really excited that George Firth will join us for this project and hopefully also take it further to PET Metallomics then. And Tom Bartnikas has kindly shared the SLC3010 knockout mouse with us. So we know that this mouse has a phenotype, so they unfortunately die at the age of 12 weeks. And have significantly reduced weight already at 8 weeks. And they have massively increased levels of manganese in the liver and the brain. So hopefully with that work, we will also sort of span our research from basic science to translation in the future. And with that, I want to thank everyone who's been involved in this work, obviously my group who are doing the work in the lab, and the continuous support by Steve Wilson's group at UCL, people involved at King's, obviously Phil Blower's lab as well, and George, the London Metallomics facility, and other people across universities in the UK and abroad. So thank you.
Thank you very much, Karen. That was a beautiful talk, and congratulations to the new things that you have discovered very recently. It looks fantastic. We still remain just upon schedule here. So either if the people who have posted questions could ask them later on today, or email them to you, or both, to be absolutely sure that they are answered, that would be great, I think. And I think we have to pass on to the next speaker. And it's my great pleasure to introduce to you Professor Michael Aschner, who is director of the Einstein Center of Toxicology at Albert Einstein College in New York. And he's going to talk to you about manganese-induced neurotoxicity, lessons from worms and human neonates. The first time I read that, I read human nematodes, but luckily it's neonates instead. A lot nicer.
Thank you. Can you hear me?
Yes, we hear you, and we can see your slides.
You can see my slides because I cannot see them. Okay.
Okay, over to you, Mickey.
Okay, thank you. For some reason, I cannot see my slides. Okay, I don't know why. That's very strange. Okay, I don't know. You can still see my slides.
We do see your slides, Mickey.
Yeah, and I cannot see my own slides on my computer.
Mickey, did you share the screen or did you share the specific window?
I went to the sharing on in Teams and clicked on my presentation.
So you might want to share the screen rather than your specific presentation. That might help if it's still a problem.
Okay.
So you can stop sharing and reshare again. See if that helps.
I don't know. Did the slide move now?
Yeah, it did.
Okay, so I can't see you, but I can see my slides. Let's go this way. Okay, so it's, first of all, thank you for inviting me. I'm actually in Dubrovnik at another meeting. I think I heard Phil before saying he's doing another meeting as well. But it's a pleasure. This is really interesting, and I watched all the talks. Very, very interesting, very innovative, and I have a very difficult feat to follow after Corinne's beautiful work. So I will try to do my best. So I will talk very little about manganese because you've heard about it already. I will talk on some work that I've done with Sham Mukhopadhyay, which some of you know, on manganese transport in HeLa cells, and the work that we did in C. elegans. And I'll show you some, at the end, some studies that are unpublished yet, which basically ask whether dopamine is really essential for the neurotoxicity of manganese and other compounds such as MPTP. And I'll describe the work for you again in the C. elegans model. So as far as manganese, you've heard about it already. I'm just going to say it's essential. I've listed some of its functions. There's a number of enzymes, and I'm not going to read them all, that are manganese dependent. So unlike some of the other metals that I work with, some of you as well, manganese is essential. Other ones are non-essential. Given the essentiality, as you'd expect, there is human intake. At a steady state, it's about 2 to 9 milligrams per day. The absorption is relatively low because of the steady state condition. So only about 3% of the manganese is absorbed in adults, and once transported, it is ubiquitously transported to all cells for the various functions that I've already mentioned. Now, Corinne mentioned some of the risks that are associated with manganese. She also mentioned that high levels of manganese are associated with a disease which mimics Parkinson's disease. The disease itself is known as manganism. And manganese tends to accumulate in motor controlling areas, predominantly in the brain, caudate putamen, globus pallidus, substantia nigra, and subthalamic nucleus. The risk, populations at risk are parenteral nutrition. We've looked at that several years ago for no reason other than the fact that maybe a little bit is good for you and a lot more should be excellent. Manganese and other trace elements are supplemented at exceedingly high concentrations in parenteral nutrition. And we've imaged some neonates on parenteral nutrition, and you can very readily identify the T2 weighted signal in MRIs. I'm not going to show any of these data. Iron deficiency is a global problem, and the reason for increased accumulation of manganese in populations that are iron deficient is that many of the transporters that you've heard about today are pretty promiscuous. So manganese and iron can compete on a number of these transporters, both in the divalent oxidation state and in the trivalent oxidation state. And at least one of the exporters for iron, ferroportin, seems to also transport manganese, some work that we've done several years ago. There are some other environmental conditions that are associated with increased deposition of manganese in various tissues, including the brain. Some are more controversial than others, but there have been studies showing behavioral deficits in children that are exposed to high levels of manganese in drinking water. This is work that was done by Regina Wasserman and her collaborators in Bangladesh. MMT seems to, it always reminds me how bad students are, we are of our history. MMT is basically a substitute for lead. It's an anti-knock agent. It's an oxide of manganese, and where it's being used in Canada and other countries, manganese has been shown to be at approximately twice the atmospheric levels than prior the use of MMT. Welding is controversial. There's many fumes coming out while welding, but one of the fumes or the metals in the fumes is manganese, and there have been studies by Brett Rossett and others showing that exposure or welding is associated with the development of Parkinsonism-like syndromes at a much earlier stage of life compared to the population at large. And Corinne mentioned methcathinone before. This is a drug of abuse. It's generated by oxidizing either ephedrine or pseudoephedrine, and the compound that's being used is potassium permanganate. Hence, the symptoms that develop, again, resembling Parkinson's disease in young people are secondary to exposure to the contaminant, which is the manganese in the potassium permanganate. So I have this slide. It's a little bit complicated, but I'll try to simplify it. The reason for the similarities between Parkinson's disease and manganism are actually very simple. In Parkinson's disease, it's the nigrostriatal pathway which is predominantly affected, which is, I don't know if you can see my cursor, but it's shown here. But if you look at this as a hose, you'll see that the area where manganese accumulates, predominantly the globus pallidus, is downstream of the striatum. There is a GABAergic projection from the striatum to the external capsule of the globus pallidus, and another one from the external capsule of the globus pallidus to the internal capsule of the globus pallidus. And if you look at it basically as a hose, think about it as a hose, it really doesn't matter whether you obstruct the water flow at this level or this level, ultimately you're going to impede the flow of the water. It's not going to come out at the end of the hose. So there are distinct differences between Parkinson's disease and manganism, and neurologists can distinguish between those, but essentially the symptoms are very similar. Now, we've been interested in manganese transporters. I'm always asked the question as to why are there so many transporters for manganese? I don't have a good answer. You would think that the level of a heavy metal, if a metal such as manganese, could be regulated by 2 or 3 importers or 2 or 3 exporters. But last I counted, there's probably about 14 importers of manganese and at least 2 exporters for manganese. And what I'm going to focus on today is only one of them, one that you've heard a little bit about, or a lot actually, by Corinne. It's the SLC3810 transporter. I'm not going to go into all the importers because we just don't have enough time. Suffice it to say, again, that many of these transporters are promiscuous. So if you look, for example, at ZIP8, ZIP14, or DMT1, you can see that in addition to manganese in the divalent oxidation state, it can also transport other cations. I mentioned manganese in the trivalent oxidation state is coming out on ferroportin, again, competition between the two. And transferrin, which was mentioned as a transporter for gallium before by Phil, can also transport manganese in the 3+ oxidation state. So many of these transporters can transport a number of metals. So the SLC3810, again, you got a good introduction, and the work that Corinne has done and some other groups, her original work in 2012. This also raised our curiosity, specifically because the patients that were described had high levels of manganese in the blood. And there were a couple different mutations that have been shown in these patients. They're listed here. One of them is missense, and one of them is a nonsense. They're shown here in the transporter in yellow and in the red star. So the work that we've done, again, together with Shamshuva Mukappadai, was basically to determine whether the mutated transporter is dysfunctional. And what you see in this slide is labeling for SLC3810 for calnexin, which is basically just a marker of the endoplasmic reticulum, and then emerge between the two. And if you look at wild type, again, these are HeLa cells, you will see that the SLC3810 seems to be predominantly, or exclusively, I should say, be deposited, is distributed on the cytoplasmic membrane. You could see it here. When you look at the different mutants, the 2 that we generated are those that have been described in patients. There are 3 other mutants. You can see that the distribution is very different. It's very diffuse, and there seems to be nothing in the membrane, which was the first hint, basically, that when mutated, this exporter is not functional. We have looked at it quantitatively, and you can see that almost 100% of the wild type transporter is in the membrane, and the different mutants are not. You can hardly find any within the membrane. We've also done ubiquitination studies, which basically tell you whether a given protein is localized to the membrane. And to do that, we use GPP130, and I'll come to this protein in just 1 minute. But GPP130 is not a cytoplasmic protein. So when you do ubiquitination studies, you can see that you cannot find it. There's no band here localizing GPP130. When we look at SLC3810, we do see a band, and when we look at one of the mutated forms, and this is true for all the others, there's no band. Again, confirming that when mutated, this transporter is not localized to the membrane. And now GPP130 is a protein, it's basically a chaperone for manganese. So when manganese levels in the cell are increased, what GPP130 does, it transports, it chaperones the manganese from the endoplasmic reticulum to lysosomes, where the protein is then degraded. What you're looking here is GPP130, and here it's in the presence of high levels of manganese, high exposure of HeLa cells to manganese. So when these cells are exposed to high levels of manganese, the red fluorescence, the GPP130, disappears. And the reason for that is that, as I said before, GPP130 chaperones the manganese to the lysosome and terminal endosomes, and the GPP130 is being degraded. Now, if you take the same cells and you first transfect them with the wild type SLC3810 in green, and then you treat them with manganese, you can see that you can preserve the fluorescence, the GPP130 fluorescence. And the reason for that is, we believe, because when you have a high complement of the wild type SLC3810, there's no requirement for the GPP130 to mobilize or to move, chaperone the manganese from the endoplasmic reticulum to the endosomes and the lysosomes. So basically, the GPP130 fluorescence is preserved. This panel here is basically identical to this one. This is in the wild type. These are the different mutants. So again, GPP130 you treat with manganese, but here we have transfected or expressed in the cells the mutated forms. And you can see that unlike with the wild type expression of SLC3810, when you express the mutated forms of SLC3810, you lose the GPP130 fluorescence. And the reason for that is that although now you have a very high complement of SLC3810 in the cell, it is dysfunctional. It does not anchor in the membrane, as I've shown you in the previous slide, and therefore the GPP130 fluorescence disappears. You still need the GPP130 to chaperone the manganese to the endosomes and the lysosomes. We've also measured manganese intracellularly. A very simple experiment. Manganese concentrations here, different concentrations. I'm sorry, the level of manganese per microgram lyset. These are the different concentrations the cells have been treated with. These are the controls, and you can see when we express the wild type SLC3810, the levels are much smaller. The reason for that is that now you have a functional transporter that is able to move the manganese from within the cell to the outside. We've done some pole chase experiments which basically confirm the same thing. Now, when we first submitted the paper, actually, we were asked to confirm that there's also some functional differences. So we did that. This was done in midbrain neurons, unlike the HeLa cells that I've described before. And we had 4 different groups. We had wild type and mutated SLC3810. In the black are just controls, and the gray bars are cells that have been treated with manganese. So if you look at wild type cells in the controls, these are different parameters that we looked at. The length of the neurites, the number of primary neurites, basically how many number of neurites are expressed per neuron, and so forth. You can see that when you take the controls and you compare them with cells that have been that express a high complement of the wild type SLC3810, there's basically no statistical significance. So the high complement of the SLC3810 is protective in essence because it is able to move the manganese from within the cell to the outside. In the absence of manganese, we don't see much damage. It's not significant in the controls, but when we treat them with manganese, now you don't have a functional mechanism to excrete the manganese from the cell, because as I said before, these transporters do not dock within the membrane. Now you see significant damage. There's loss of neurite length, there's loss of the total number of primary neurites per neuron, and the other parameters. You can see each of these look at all the different graphs here look basically identical. So having SLC3810 is protective to the cells, likely because manganese can be removed extracellularly. But if you express one of the mutant forms, you are unable to protect the cells because these transporters, we believe, are non-functional. Now, we've gone on to replicate some of these studies in C. elegans. I'm not going to say very much about the model. Suffice it to say that they only have 959 neurons. And for those of us who are interested, 302 neurons out of 959 cells. And for those of us interested in dopamine, there's only 8 dopaminergic neurons. We've looked for SLC3810 in the worm. We looked for a homolog. We found a few, but the highest sequence identity was 38%. The function was unknown. And given the low sequence identity, what we decided to do is actually express human SLC3810 DNA in the worms. I'm going to show you first a movie on the dopaminergic neurons, because we're going to look at them later and see whether they degenerate. And basically what we have done here is to put a green fluorescent protein on the promoter for the dopamine transporter. The dopamine transporter is expressed only in dopaminergic neurons. And you can see there are 6 dopamine neurons in the head, and there's a couple of them in the tail. They show up once in a while. You'll have it right here, right now. So you can visualize these. You can do the same thing with any other type of neuron. And you can assess neurodegeneration. Let's see if it works. So we've done that. And we've used different kinds of strains of C. elegans, strains that express green fluorescent protein in GABAergic neurons, cholinergic neurons, or chemosensory neurons. In this case, it's red. And these are worms that are not treated with manganese. These are manganese-treated worms. And the only neurons actually that degenerate in the presence of manganese are the dopaminergic neurons. We found no change in the cholinergic neurons, in the GABAergic neurons, or in the chemosensory neurons. And this is shown here. As you increase the concentration of manganese, you can see that the axons basically start to lose fluorescence. And eventually, as you treat with higher concentrations of manganese, there's even decreased fluorescence in the cell bodies of these neurons. Now we can look at the neurodegeneration. In this case, we have 3 different strains. We have the controls. We have the worms that have been where we express the wild type SLC3810. And we have worms where we expressed the mutant, or one of the mutant forms, the L89P of SLC3810. This is without manganese. This is in the presence of manganese. And you can see that when you treat with manganese, there is about 50% degeneration of dopaminergic neurons, even in the controls. In the wild type neurons, in the neurons that have been transfected or were made to express the SLC3810 wild type isoform, there is some rescue. It's by no means complete. It doesn't go all the way up, but we don't see this rescue in the worms that express one of the mutant forms of SLC3810. We can also perform functional studies in worms. This is known as basal slowing response behavior. The worms are very similar to us when you present them with food. They slow down. So you can count the number of body bends per 20 seconds in the absence of food and in the presence of food. And there's a delta between no food plus food, and it's about 8 to 10 bends per 20 seconds. So what you're seeing here is the delta, the difference. This behavior, the slowing down, the basal slowing response, is driven by dopamine. If you look at worms, the 3 different strains, but in the absence of manganese, they look the same. If you look at the strains with manganese, you could see that this very much mimics the neurodegeneration. It's a very good functional readout of dopaminergic neurodegeneration. Again, there's inability to slow down. The delta is smaller in the presence of manganese. It goes from 10 to about 4. But if you express the SLC3810 wild type isoform, you can recover this behavior. Again, it's not identical to the control, but very close. But you don't see this kind of recovery when you express the L89P mutant form of the SLC3810. So basically, we think that the SLC3810 wild type, as you would predict, docks to the membrane. We don't think the mutant form can dock within the membrane. We don't know exactly where it is, but the fact that manganese concentrations in the cell, in the worm, increase is related to the fact that you hinder the export of manganese by way of this transporter. Now I'm going to talk for 4 minutes, 5 minutes, on the role of dopamine in neurodegeneration. These are some very preliminary studies, but I wanted to share them with you. And basically, we are asking the question as to whether dopamine is really essential for the neurodegeneration that you see with the manganese and with the MPTP, which is basically a designer drug. I'm not going to have time to talk about it, but it does target dopaminergic neurons. So what this scheme shows you is the synthesis and catabolism of dopamine and serotonin. On the left is dopamine, on the right is serotonin. You can see the nomenclature for the human genes and the C. elegans genes for both of these neurotransmitters. And if you compare them, what you'll notice is that there's only 2 differences between dopamine and serotonin neurons. One of them is that in worms, you need CAT2, or the mammalian homolog tyrosine hydroxylase, to make the dopamine. In C. elegans and in humans, you need to use tryptophan hydroxylase to make the serotonin. These are exactly the same. And then for the reuptake of serotonin, the SERTs are referred to as mode 5. For the serotonin neuron, and for dopamine, it's DAT1. I've shown you before the movie of the DAT1 tagged worms with the green fluorescent protein. So what we basically decided to do is to ask, does dopamine really, is it the culprit in the neurodegeneration? And to do so, we decided to take serotonergic neurons to knock out TPH, MOD5, and express CAT2 and DAT1. And we call these pseudo-dopaminergic neurons. So these neurons now, instead of making serotonin, they are making dopamine. These are the neurons. Again, I don't have enough time. The ADF neurons are serotonergic neurons, and these are the wild type. They express TPH and mode 5. What you're seeing here is dopamine staining for worms. The BY200 are the dopaminergic neurons, the strain that expresses green fluorescent protein in dopaminergic neurons. There are 6 neurons in the head. We use the formaldehyde-induced fluorescence, and you can see here the blue. This is the dopamine. And if you overlay these 2, you can see that they are expressed in the same cells. Again, you'll have to believe me that there are 6 of these neurons, the CYPs and the ADEs, in the head of the worm. This shows what happens in the presence of manganese. These again are wild type worms, and when you treat them with manganese, the blue disappears, so you get neurodegeneration of dopaminergic neurons. And what you have here is now ADF neurons that express that, okay, the same thing as these do, but these are serotonergic neurons. And you can see that they also express, they also make the dopamine, the synthesized dopamine. When you combine these two, the serotonergic neurons now make dopamine. They're pseudo-dopaminergic neurons. Now we've tested this up to this point with MPTP, with 6-hydroxy dopamine. We're doing it right now with manganese. Looking here at serotonergic neurons, ADF neurons, wild type neurons that are treated with MPTP, again, a poison, a toxin for dopaminergic neurons. You see no neurodegeneration. In pseudo-dopaminergic neurons, so serotonergic neurons that are made to express the dopamine, you see significant neurodegeneration. Another way to ask this question is to basically knock down in dopaminergic neurons CAT2, which is tyrosine hydroxylase, or DAT1, or to knock the two of them combined. So here we have 4 different groups. These are the wild type worms without manganese, the CAT2 knockouts, the DAT1 knockouts, and a combined knockout. In the absence of manganese, there is no difference in dopaminergic neurodegeneration. In wild types, if you treat them with manganese, there's about 50% neurodegeneration, consistent with what I've shown you before. But if you knock out CAT2 or DAT1, they cannot make dopamine, they cannot reuptake the dopamine. If you have a double mutant, they cannot make it, they cannot take it up, and you completely protect these neurons from the toxicity of MPTP, and you're basically seeing the same thing with manganese as well. So in summary of this part, we have now engineered 5-HT or serotonergic neurons, ADF neurons, to make dopamine, where we can study the role of dopamine in neurodegeneration. Unlike the serotonergic neurons, which are not sensitive to manganese or MPTP, when you make them to synthesize dopamine, they become sensitive to a number of these compounds, MPTP, 6-hydroxydopamine, which I didn't show you, and manganese. And with this, I'll stop. I want to thank a number of people in the lab, Pension, Julia Bornhorst, and Mahfouz, who have done the work. The last part is being done by Mahfouz, who's a PhD student, my longtime collaborator, Aaron Bowman, as well as the folks at the University of Texas, with which we have done the work on the HeLa cells. And with that, I'll stop. I think I'm on time, and I don't know if there's time for questions. If there is, I'm happy to answer them. Thank you very much.
Thank you very much, Mickey. It was really nice. I loved your wondrous, miraculous beasts.
Thank you.
That you have. Yeah. Regards to questions, as people know, there's a Q&A window. So perhaps the easiest way of answering questions will be for Karen and Mickey to go to that window and then answer directly to those questions. And then also everyone can see them, as opposed to emails being sent out and it being done behind the scenes. So we now have a 15 or a 12 minutes break until Puwa Su is going to talk about the iron killer. And so, yeah, so make the most out of that. Make your coffees, and we'll see ourselves in 12 minutes again.
Right. Hello. Hi, everyone. Welcome back. My name, if you forgot, is Giovanni Mann, and I'm the chair for this session this afternoon. But I thought before I introduce my colleague Po Wasu, I would do some more housekeeping. We've got a lot of questions that are going into the Q&A box, which are fantastic. And I think if the speakers that spoke would access that question box, they can actually reply, and then everyone can see answers. I initially started emailing some of the speakers this morning, but those emails are personal. They weren't meant to be personal, but I think if the speakers could try to go to the Q&A box, that would be really helpful. The other thing that I wanted to say is that the web page that's there now, the holding page that Richard Xiao put there, who I'd like to personally, before I forget at the end, thank for putting together all of this elegant King's webpage, the program, the whole show. I mean, really, without you, Richard, we wouldn't have had this as elegant as today. But there is also a full detailed abstract booklet that you can download from this page that you see just now. And I do recommend you do it. It has all the contact details, and importantly, it also has the details of all of our sponsors for the London Metallomics facility. And they, I believe, are tuning in today too, because it's in their interest and our interest to maintain our very fruitful collaboration. So that's my monologue completed. Welcome back again to those who have just joined. It's a personal pleasure to introduce Dr. Po Wasu from the Institute of Psychiatry, Psychology and Neuroscience at King's. And when we were talking about her title, she came up with Iron, Star Killer Strikes Again. So I'm looking forward, Poa, to your interesting lecture. Thank you.
Thank you, Giovanni. Thank you for your kind introduction. I will try and share my slides. Okay, that's interesting. Right.
Poa, have you got your slides loaded?
I do, but I'm trying to use one screen because I felt that it might be better, but I'm experiencing slight technical difficulty.
Okay.
Hold on. No, I think I've got it now. Hope you can see it.
Yes, we can. If you just make it large, brilliant.
Yeah, I'm going to project it. Is it present? There you go. So, hello everyone. Thank you, Giovanni, for the introduction, and also thank you to the organisers for inviting me to speak today about my lab's work on iron in brain aging and Alzheimer's disease. So let's take a quick trip to the sun. Careful now, don't get too close. Atoms of hydrogen being smashed together into helium with a release of energy that prevents the sun from collapsing on itself. And fusion reactions are ongoing for billions of years, so we don't have to worry about this, with the production of heavier metal elements until we get to the 26th element, iron. Now, energy is required to fuse iron, and as energy is not produced anymore, the star's bigger than our sun, the star goes supernova, spewing its contents across the cosmos. So, by the way, I had a rather sensationalist title saying that iron kills stars, but actually iron doesn't poison stars. It's really comparable to the ash that you get from a fire. So, moving back to this star material or dust, that gets incorporated into everything, including us. And if you don't remember anything from today, that you don't remember from my talk, please remember you are star material. The iron that was formed in the star prior to supernova is in trace amounts in our body. So let's go back to Earth. While only in trace amounts, iron is crucial for life. Iron is needed for many life-sustaining processes, including mitochondrial respiration, oxygen transport, and specifically in the brain, iron is vital for neurotransmitter synthesis and myelination. Hopefully you can see my cursor here. But of course, with the good, there's always the bad, right? The bad is that ferrous iron, or Fe2+, can react with hydrogen peroxide. There you go, that's my cursor. From mitochondrial respiration to oxidize Fe2+ to Fe3+. And that's not an issue, but the problem is this formation of the hydroxyl free radical. And, oh, I think I've lost my, there we go. The problem is that the hydroxyl free radical is very reactive and will react with surrounding molecules to produce reactive oxygen species. Oops, I'm going backwards. There we go. And lead to oxidative stress and eventually cell death. Now, it's difficult to avoid the toxicity as hydrogen peroxide is continuously produced as a byproduct of mitochondrial respiration. So one way for cells to cope with this is to avoid iron toxicity is to regulate their iron content. Iron enter the cells via the transferrin receptor and DMT1, and the iron enters the labile iron pool, and this labile iron pool is a dynamic pool of redox active iron that provides iron for cellular processes, but can undergo Benton reactions, and so it's potentially toxic, and the labile iron pool has to be regulated. So excess iron can be transported out of the cell by ferroportin, assisted by ceruloplasmin, which oxidizes Fe2+ to Fe3+. Ferroportin is the only known iron cell exporter, and it's downregulated by hepcidin, which causes the ferroportin to be internalized and degraded. And the net effect is that you have iron retention. Now, rather than be exported, excess iron can be stored safely in ferritin. So in this way, it's stored safely inside the cell and can be readily available when the cell needs the iron. And this pool, this labile iron pool, is regulated by iron regulatory protein 1 and 2. And these proteins regulate the pool by regulating expression of the iron importer, exporter, and storage proteins. Now, when we're considering regulation of iron within the brain, so as a whole, rather than just in cells, iron is also regulated, brain iron is also regulated by the blood-brain barrier and the blood-CSF barrier. So despite regulation, brain iron does accumulate with aging. And that's what I've shown here. So this is from a Holgren and Saranda paper in 1958, where they took tissues from different brain regions, extracted them, and did a spectrophotometric analysis. And you can see that there's a rapid accumulation of iron from birth, and then it starts to plateau. And you see a similar picture in the various brain regions, although the amount of iron that's deposited vary between regions, with greater amounts of iron being deposited in the basal ganglia regions compared to cortical regions. So the question for us was, do we see this age-associated brain iron accumulation in normal mice? We use synchrotron radiation X-ray fluorescence to quantify, map iron throughout the mouse brain at different ages. So we did our iron mapping at the diamond light source. Brain sections were scanned with a spectrum from each beam position that was deconvolved to produce these type of maps. So I've shown you typical iron and zinc maps from human brain tissue with white matter containing higher amounts of iron and zinc. I'm not going to talk about this data today. That was presented, actually, some of this was presented by my PhD student who did this about an hour ago at an MRI conference that we're currently at as well. But if you invite me again next time, I'll talk about this. So back to our mouse aging study. We use synchrotron radiation XRF to look at iron accumulation with aging. And we see that that happens similar to that in humans. And again, especially in the basal ganglia regions. So I've shown the striatum, globus pallidus, and substantia nigra. And if we quantify the data, we see that there's significantly higher amounts of iron at 19 and 27 months of age in mouse brains in the striatum and globus pallidus. So we'll say that mice can be used to model age-associated brain iron accumulation. Why is this interesting? Well, if you go and look at that paper, you'll see that we used it to have a look at the effects of a anti-brain aging regimen in mice. But actually, I've gone a little bit ahead of myself because I wanted to take you aside a bit to actually talk about iron and MRI. And that's because, for those of you that know me very well, will know that I come from the magnetic resonance field. And I've been developing and applying MRI, MRS, and NMR and biomedicine. And I became interested in iron as I was using iron nanoparticles to develop MRI-based molecular imaging methods, since MRI is sensitive to the presence of iron, as shown here in these two panels. So these are gels doped with different concentrations of iron chloride and so ferrous chloride and ferric chloride, so Fe2 plus and Fe3 plus. And I hope you can see that the MRI signal decreases with increasing iron concentration. So at 5 millimolar ferric chloride, we can barely see a signal from the gel. And if we were to do a basic spin echo MRI experiment and we vary a parameter called the TE, the echo time, we get a series of curves at different concentrations, iron concentrations, from which we can measure a relaxation time constant, or T2. And if we take the reciprocal of T2, we get the relaxation rate constant, or R2. And if we plot the R2 against the iron concentrations, we get these straight lines. And these lines allows us to compare the effect of different iron forms on the MRI signal. And the steeper the line, the more effective it is at modulating the MRI signal. So ferric chloride is better than ferrous chloride. Note that MRI does not directly measure iron. It doesn't directly detect iron, but the effect of iron on the surrounding water molecules. And so that's how we I got into iron mapping using non-MRI methods. So for example, electron microscopy, histochemical staining. I've mentioned about synchrotron radiation XRF, and you would have heard about laser ablation ICP-MS, which is available in the LMF. Just to mention that these non-MRI methods are not able to assess iron in the living brain, and so the need for MRI for clinical translation. So MRI has been used to look and to assess brain iron accumulation in the aging brain, and I've shown some of the data here. So remember back to this measurement called T2, this relaxation time constant that we can get with MRI. We see that the T2 value decreases with aging, and then it plateaus, and that mirrors the increase in iron and the plateauing that we saw earlier in previous slides. There is also another parameter that's similar to T2 called T2 star, and people have published to show that T2 star also decreases with age, suggesting decreases with increasing age, suggesting that there's an increase in iron with aging. And so if we go back to our study, we actually did, I need to remember to say this. So sources of MRI contrast. So you realize I've just said that iron can modulate our MRI signal, but other things can also do that, including myelin and cell density. And so that's why we're always doing using corroborative methods if we can. And this relating our MRI signal with biological or metal measures is an area of active research by my lab and others. So yes, we'll go back now to our mouse brain study, and you'll see that we've got our XRF iron map here, but before we obtained that, we actually took the brains and did ex vivo MRI to give us these R2 maps, and then we sectioned the brains for XRF. So we could correlate our R2 values with our iron levels, and we can see there's a good correlation between R2 star and iron, and so that's consistent with us wanting to use R2 star as a non-invasive measurement of iron in live organisms. And so this was actually ex vivo data that I'm showing here, but in the paper there is in vivo data, but this data does show us that R2 star increases, which we propose is due to increases in iron with aging in various basal ganglia regions. So we are particularly interested in age-associated brain iron accumulation, as we hypothesize that this is one of the factors that contributes to the aged brain to developing neurodegenerative diseases, because indeed advanced aging is the major risk factor for neurodegenerative diseases such as Alzheimer's disease, and indeed there have been reports that iron is even higher in the brain in neurodegenerative disease. So I'm now going to focus on Alzheimer's disease. Alzheimer's disease is a chronic disease that slowly kills neurons and leads to cognitive disability, including memory loss, and it is the most common form of dementia. A diagnosis of Alzheimer's disease is only confirmed at postmortem by the presence of amyloid plaques and neurofibrillary tangles. Now, amyloid plaques are extracellular aggregates of beta amyloid protein, and neurofibrillary tangles are intraneuronal aggregates of abnormally hyperphosphorylated tau protein. It's been suggested that removing amyloid plaques can successfully treat Alzheimer's, but anti-amyloid clinical trials have been largely unsuccessful. One of the issues with amyloid being the cause of Alzheimer's disease, so this amyloid cascade hypothesis, is that beta amyloid accumulates in the brain over 30 years before cognitive deficits are observed. There's not a good correlation between plaque deposition and cognition. And not all individuals with a high beta amyloid load develops Alzheimer's disease. So some people have lots of this amyloid plaque in their brains, but they do not have Alzheimer's disease. Although, to clarify, that all Alzheimer's patients obviously will have a high brain amyloid, since amyloid is a hallmark of only found in Alzheimer's. So there's a real push to explore other avenues, including the possible role of iron homeostasis in Alzheimer's disease. Why iron? Well, there are many lab and clinical studies and clinical trials that have suggested iron dyshomeostasis in Alzheimer's disease. For example, iron is elevated, especially in the basal ganglia regions of Alzheimer's disease. A recent paper, well, it's 2021, showed that iron was increased in the inferior temporal cortex of Alzheimer's patients with confirmed amyloid and tau pathology, but iron was not, did not change in those individuals with amyloid and tau pathology, but not clinically diagnosed with Alzheimer's disease. And so to lab studies, iron has been shown to bind to and increase amyloid plaque and neurofibrillary tangle formation. And it's thought that they may actually provide sites for redox reactions that could lead to the production of damaging reactive oxygen species. What's also interesting that in Alzheimer's disease, many of the iron regulatory proteins are altered. We ourselves saw a decrease in the CSF melanotransferrin levels in mild cognitively impaired individuals that progressed on to develop Alzheimer's disease. And the whole field has been revitalized by the discovery of ferroptosis. Now, ferroptosis is a type of regulated cell death that is morphologically, biochemically, and genetically distinguished from other forms of programmed cell death, including apoptosis. A similar type of cell death had been previously reported, called oxytosis, but this doesn't place any focus on possible involvement in iron, although it's thought that maybe the oxytosis and ferroptosis pathways may be one and the same. For us, what's exciting is that ferroptosis allows us to directly relate iron dyshomeostasis to neuronal death, and so clinical symptoms of Alzheimer's disease. So ferroptosis, as I said, is regulated cell death, and it results from the accumulation of lethal phospholipid peroxides and insufficiency of cellular antioxidant systems, especially glutathione peroxidase 4, GPX4, which catalyzes the reduction of lipid peroxides to its corresponding less toxic alcohol. Lipophilic antioxidants, such as vitamin E, can be anti-ferroptotic by inhibiting lipid peroxidation. And there's been reports that an endogenous metabolite of vitamin E has demonstrated potent anti-ferroptotic activity. Now, while significantly elevated iron levels are not required for ferroptosis, higher iron increases the susceptibility to ferroptosis by enhancing lipid peroxidation. This can be by non-enzymic means, so via the Fenton reaction, or enzymatically by lipoxygenase catalysis. Lipoxygenases are iron-containing proteins that catalyze the oxygenation of polyunsaturated fatty acids, or PUFAs, to form hydroperoxides that contribute to lipid peroxidation. The importance of iron-enhanced lipid peroxidation in ferroptosis is underlined by the anti-ferroptotic effects of iron chelators. It's worth noting that iron chelators have been shown to attenuate cognitive decline in Alzheimer's patients and in animal models. So we undertook a study to look for evidence of ferroptosis in post-mortem human brain tissue samples from the temporal gyrus, a brain region affected in Alzheimer's disease, hypothesizing that characteristics of ferroptosis, so iron dyshomeostasis, augmented lipid peroxidation, and impaired glutathione antioxidation would be observed in Alzheimer's disease. We also conjectured that interactions between proteins, elements, metals, and metabolite networks differed between control and Alzheimer's brains. So there have been studies published looking at these aspects in Alzheimer's disease, but separately, and to the best of our knowledge, our study was probably the first to look at these aspects simultaneously in post-mortem human brain tissue from Alzheimer's disease. So our multidisciplinary study approach, which we think is quite unique, is to look at levels of proteins using Western blotting. So iron proteins involved in iron regulation, glutathione antioxidation, and the levels of 4-hydroxynonenal adducts, which result from lipid peroxidation. We use TXRF, that's total reflection X-ray fluorescence spectroscopy, to measure transition metals and elements. And we've previously used this technology to look at differences between in plasma between plasma from control and Alzheimer's patients. And we also use untargeted NMR-based metabolomic analysis of both water and fat-soluble metabolites too. Now, you remember that ferroptosis is lethal cell death induced by lipid peroxidation. Reactive aldehydes, such as 4-hydroxynonenal, is a well-known byproduct of lipid peroxidation. And we see that hydroxynonenal adducts are significantly increased in Alzheimer's disease. So this is consistent with the many reports of increased lipid peroxidation in Alzheimer's. Now, plasma membrane polyunsaturated fatty acids, or PUFAs, are particularly susceptible to ferroptosis. And we saw there was a trend of lower PUFAs in Alzheimer's, suggesting increased PUFA peroxidation. We also measured levels of this protein ACSL4, and what this enzyme does is to insert arachidonic acid, that's a PUFA, into phospholipids, especially phosphatidylethanolamine, and determine the lipid composition of plasma membranes. So the greater the content of PUFAs incorporated into plasma membranes, the greater the susceptibility to lipid peroxidation and ferroptosis. Now, we saw that the ACSL4 levels were similar in control and Alzheimer's disease, but what was interesting was that it was negatively correlated with GPX4 in Alzheimer's disease, suggesting that increased ACSL4 is associated with decreased GPX4 function, possibly resulting from enhanced lipid peroxidation of plasma membrane in Alzheimer's, and that in turn can detoxify the GPX4. I can see Giovanni is waving at me. Okay, I need to crack on and talk about iron dyshomeostasis in this tissue. So using TXRF, we saw that levels of iron were comparable in birth control and Alzheimer's disease. But what was really interesting was that we saw a significant increase in the light and heavy chain ferritin, which suggests that there is a functional iron overload in Alzheimer's. And just to say, ferritin is made up of light and heavy chains. So while ferritin is normally, while iron is normally stored in ferritin in a non-toxic and bioavailable form, there is experimental evidence to suggest that ferritin is abnormal, and it may ferritin in Alzheimer's may lose its ability to maintain iron homeostasis and may provide additional sites for detrimental redox reactions and enhanced oxidative stress. So I'm going to try and take you through this quickly. So there's a negative association between ferritin light chain and GPX4, and that is in Alzheimer's and not seen in controls, again consistent with dysfunctional ferritin in Alzheimer's, negatively modulating GPX4. And elevated ferritin may increase ferritinopathy, which can lead to increased release of iron to promote lipid peroxidation. And what's interesting is ferritinopathy is required for the induction of ferroptosis by a bromodomain containing protein 4 inhibitor. So there's an increase in ceruloplasmin in Alzheimer's disease, and we think that's a compensatory increase to aid ferroportin export of iron from the cell, although we only saw a trend of a decrease ferroportin. What was interesting, although we saw similar levels of transferrin receptor and DMT1 in both control and Alzheimer's brains, was that there was, from correlation analysis, that iron was positively correlated with the transferrin receptor in Alzheimer's. It's not what we expect physiologically. If you have high iron, you should have decreased expression of the transferrin receptor. So we suggest that there is iron dyshomeostasis in Alzheimer's. And what's particularly interesting is that transferrin receptor has been proposed as a possible marker of ferroptosis. So I can see definitely Giovanni is signaling at me. So I just wanted to say quickly about glutathione antioxidation, and actually there's a lot to say about this, but I'm going to try and summarize it a bit more. Is that we saw there was a decrease in
yes. Giovanni, you have 1 more minute, please.
Okay, so I shall try and see what's important here. So we see an increase in XCT, which suggests that there could be glutamate exocytotoxicity because it actually transports glutamate out of the cell and brings in cysteine. I'm not going to say any more about that, but it's a very, very interesting story. And we did see an increase in the glutamate to GABA ratio in Alzheimer's, which is consistent with the exocytotoxicity that that's been reported in Alzheimer's disease. I'm not going to talk about the glutamine metabolism. That's also a very interesting story, but we did see a decrease in glutamine, and glutamine has anti-ferroptotic effects. I'm not going to explain more about it, but it just shows how ferroptosis involves lipid metabolism, protein metabolism, as well as glutathione metabolism, as well as amino acid metabolism. So, and I throw in the slide on just to show the graph for N-acetyl-aspartate. SMR people always want to look at N-acetyl-aspartate because it's a neuronal marker found by MRS, and as we expected, there's decrease in Alzheimer's disease. So I'm not going to summarize what I've said. I hope you will remember it all. But I just want to say that I did have a rather sensational title. Well, you may have realised that iron is not toxic to stars. Iron is like throwing ash on a fire. But for Alzheimer's disease, we suggest that iron is like throwing petrol on a smouldering fire. Final slide. Want to thank the people that did this work, especially Ashar, Ashraf, and also our funders. They're here. So thank you. I'm sorry I overran a bit. It's Giovanni.
That's
thank
okay.
you everybody for your attention.
Thank you very much, Powa. And I'm sure I put a question into the question and answers. Others can do the same. And then, Powa, you can answer them. It now falls upon me to welcome our next speaker, Professor Joanna Collingwood, who's Head of Trace Metals in Medicine Laboratory at the University of Warwick. And you're going to be talking to us sorry, I'm reading my own title, excuse me. The role of iron in neurodegenerative disorders, insights and opportunities with synchrotron light. Joanna, the floor is yours.
Great, thank you very much. It's fantastic to be here today. I've brought my slides into play. Can you confirm that you can see those clearly now?
We can see everything perfectly.
Fantastic. Okay, so today we're going to be looking, it's fantastic following on from Po's talk, because that's really covered all kinds of fundamentals that I didn't have time to cover in my talk. So I think this has been planned really well by the organisers. Thank you. And thank you, Po, for that introduction as well to the chemistry behind the iron and many of the iron biology features that really matters. What I'm going to look at here today are the analytical approaches, the reason why we're using synchrotron light, and the majority of the light produced by synchrotron facilities is in the X-ray part of the spectrum. So, by way of introduction, I lead the Trace Metals in Medicine Laboratory in the School of Engineering at University of Warwick, and I established this lab in order to enable interdisciplinary work, including with human tissue samples. Much of what we do in practice is to prepare samples for measurements at external facilities, including synchrotron analysis, and also work with high-resolution magnetic resonance imaging using our Brooker microimaging probe on the Warwick campus. So I'd like to start by reflecting on some things that have already been covered in the meeting today, some of the clinically observed aspects. We know that certain neurodegenerative disorders are associated with a change in metal metabolism that's significant enough to be detectable in the clinic. So for the example of Wilson's disease that we heard about earlier, there's clear evidence of copper accumulation in certain tissues, including in the cornea of the eye. And we know that metal accumulation isn't simply a benign byproduct of a change in metabolism, that metals that accumulate in Wilson's, Parkinson's and others, they're associated with neurotoxicity, and they may be a factor in disease progression. And in neurodegenerative disorders, we are by definition looking at degenerative processes affecting the nervous system, which will result in atrophy. So if we're going to look for these changes, volumetric change can be used as a marker of disease. However, changes in transition metal metabolism may precede atrophy in many of these disorders, so there's motivation to image and quantify these elements if they're markers of a change, maybe even giving some insights in disease mechanisms at a point before the patient experiences significant loss of function. And so since the evolution of clinical magnetic resonance imaging in the 1980s, it was observed that some MRI parameters are affected by tissue iron concentration, and it's that non-heme mineralized fraction of iron that's the primary source of iron contrast in tissue for MRI. And one of the most promising approaches with MRI is quantitative susceptibility mapping, and that's been advanced by a number of groups, and I've put an example here from Christian Langhammer and colleagues where they've been looking at ways to advance this technique. Now, there are certain rare disorders, including forms of neurodegeneration with brain iron accumulation, that show a signal change termed the eye of the tiger sign in one of the regions that's normally high in iron. And that shows excess iron accumulation, arguably. It's a difficult one because it's very difficult to know precisely what's changed in the MR signal. But this has been a hypothesis for some time, and there's also atrophy in this rare disorder. But it was recently reported that although it can be a sensitive marker for this disorder, it may not be specific to this. And so it's the importance of both sensitivity and specificity if imaging methods are to be used to assist the diagnosis. And in the more prevalent disorders, such as Alzheimer's and Parkinson's disease, it's not yet fully established that we can make use of changes in transition metals for diagnosis, but meta-analysis have confirmed altered tissue iron concentration in some brain regions, so putamen in Alzheimer's disease in particular. And there's been a lot of work on Parkinson's disease as well to see whether changes in iron in certain regions of the brain can be used as a diagnostic tool. So there's a recent meta-analysis for Parkinson's from May 2020 supporting the idea that there's really useful signal to distinguish Parkinson's from healthy controls. So there's lots of opportunity here, but also some analytical challenges. And in the talk today, I'm going to look at how we're addressing some of these challenges by looking at what's present in brain tissue and why we include synchrotron X-ray analysis in our work. So MRI is not the only clinically viable method that can be used to detect transition metals, but it is the most versatile and it's uncommonly used. And so in this published review, they highlight a number of challenges in the context of imaging metals, including iron and manganese and copper. And I wanted to draw your attention to some of these key points, because firstly, when we're looking at MRI signal contrast, unless we have supporting histology, we don't know for sure what the source of that contrast was. And secondly, when we look at an individual where a particular element is present in excess or in deficiency in a given region of the brain, this may be impacting other elements too, because metabolic pathways are in many cases codependent, and that's another factor that we've been hearing about today. So being able to see the spectrum of change is important. And lastly, even if we know which metal ion we're looking at, the magnetic properties are going to depend on the oxidation state and on the mineral phase. And there are small but potentially significant differences in these parameters, not only between compartments in the brain, but also between healthy and diseased states. So I'm going to show you how we're addressing some of these challenges through the use of synchrotron X-ray facilities to look at post-mortem tissue. So we know that metals are essential to normal brain function. We've heard a lot about that today. They have many roles in fundamental processes. And here I've just illustrated from a recent review the heterogeneity of the distribution of many of these metals. So most transition metal species are bound to low molecular weight species, such as the iron transport protein transferrin, illustrated here. And mineralized forms are also present. Significant proportion of iron is present as iron oxide, primarily in the ferritin protein, which incorporates a ferrihydride-like iron oxide core with a diameter of up to 8 nanometers. And in the brain, the patterns of distribution are highly heterogeneous, so we have marked differences in tissue iron concentration between one region and another. And historically, histological staining has been used to reveal metal ion distributions in tissues. However, this approach doesn't lend itself to quantitative analysis, and one of the limitations is that because metal ions are stored in a variety of ways, they're not all equally available to be stained. So in the example shown here, there's much stronger iron staining in the basal ganglia and grey matter. There's very little in the white matter. But if you take representative samples from the tissue and you dissolve it and perform a quantitative concentration analysis, you'll find some regions where levels of iron in white matter are equivalent to some of the grey matter regions. So one of the reasons that iron is so useful is the way in which it can react with other species in vivo. And that's something that we've been hearing about today in some detail. So I think here I'm not going to talk about this in any depth, but it is worth acknowledging that the production of reactive oxygen species is actually essential. It's a normal part of metabolism, but it's when we have an imbalance that we're looking at potential damage occurring. So oxidative stress is observed as one of the mechanisms of damage in neurodegenerative disorders. And the way that different regions are affected and metals are affected appears to vary with patterns between different disorders. This was illustrated some time back by Thompson and colleagues. And it's not just about the concentration of an element in a particular compartment. It's very much about the way that that element is being trafficked, transported, and utilized in processes. So among the transition metals, iron is both the most abundant in the human brain and the one where a significant fraction of the iron is present in mineralized form. And of this fraction, which we can consider to be a primary source of iron-mediated contrast in MR, we can expect the majority of this to be in ferritin, the main iron storage protein in human tissue in the human brain. And this would be antiferromagnetic at room temperature, the ferrihydrite-like oxide that we'd normally expect to see. And other mineral phases, they're present in trace quantities by comparison, but of particular relevance to magnetic resonance imaging are magnetite and maghemite, both of which are ferrimagnetic with comparatively high saturation magnetization. And we published a book chapter looking at this in some ways a very niche area, but a fascinating one a few years ago. That's in Damien Favor's book on the iron oxides from nature to applications. So when we look at what's normally in tissue, I think the important thing to recognize is its heterogeneity, but also that when we're looking at neurodegenerative disorders, we're seeing changes in terms of concentrations increasing or decreasing, altered chemical states, altered mineral phases. This is often really small change as an overall fraction of the whole, but over time, these changes can be significant. And the way in which they change and pattern changes in different diseases is very much an active area of research. And this is not an exhaustive list that I've provided here. It's just to give you a flavour of the scale of the change and the fact this is across this spectrum of disorders. So I'm going to show you initially some early work that I contributed to in a study led by University of Newcastle in the UK, looking at a region in the Parkinson's disease brain, which is particularly vulnerable to cell loss. And in the dopamine-producing neurons, we showed that the iron concentration, as determined by electron microprobe analysis, was significantly higher in the Parkinson's disease cells than in healthy controls and than in another movement disorder, in the case of Huntington's disease. And so what's interesting here is that there is a robust increase in the amount of iron in these individual cells compared to the healthy cells, but we don't see any information about what's changed, whether the iron is bound in its normal form, its normal local environment, just more of it there, or has the storage form changed? So those kinds of questions are still open at the end of that initial observation, and finding ways of looking to understand what's present in the tissue in order for it to be imaged has been the main focus of our work over the past decade or so. And so much of what we do is post-mortem work with synchrotron facilities, where we're looking at the signal that we can obtain from metals in tissues without having to do any staining or processing of those tissues beyond cutting thin sections of them. And so what we're doing in this example here on the right, trust the laser pointer here to work. So this example over here, we're working in the higher energy hard X-ray region of the spectrum to obtain maps of iron intensity, using X-ray fluorescence that's been stimulated for emission from tissues with a very high energy, very high intensity, monochromatically focused X-ray beam. And so what we're getting is multi-element fluorescence here. We're getting a spectrum of all the peaks, all of the elements that have fluorescence emission in this energy range can be detected. So we get the transition metals here, lighter elements here, and then we've also got scatter from the primary beam. And this is a really sensitive method so that we can pick up trace elements such as manganese, as well as more abundant elements such as iron. And as well as being able to collect intensity distribution maps with fluorescence to get an idea of that concentration distribution, we also have the opportunity to see how these iron foci are going to absorb the X-ray beam. And when we do that, we get information that, about speciation that allows us to interrogate the oxidation state and the local environment, including determining mineral phases where they're present. So we can work at both microscale and nanoscale spatial resolution, depending on the beamline. And the example I'm showing you here is from the I-18 microfocus beamline at the Diamond Light Source in Oxfordshire. So here's one of the things we've been particularly interested to look at, and it's about the form of iron oxides associated with Alzheimer's disease pathology. Here we're looking specifically at amyloid plaque pathology as referred to by Poe in the previous talk. I'm not going to comment on neurofibrillary tangles today, but that's obviously another key area to be looked at. So here in our original study, we've made measurements of the iron absorption spectra from iron-rich regions in Alzheimer's brain tissue using a hard X-ray microfocus beam at the Advanced Photon Source in Chicago. And we found spectral components that were not consistent with ferrihydrite. So instead, some of the spectra showed evidence for magnetite, and these spectra were observed in an amyloid plaque-rich region of Alzheimer's tissue. And we didn't have information to tell us if we were directly on the Alzheimer's plaque, but we went on to look at isolated amyloid plaque cores in collaboration with George Perry's group from UTSA. And by doing that and looking with electron beam methods, including diffraction, as shown, and also tomography, we were able to identify magnetite as a primary phase associated with iron oxides in these amyloid plaque cores. And the size distribution of them was consistent with them having originated in ferritin. So we were really curious to know from these two independent sets of results whether there was a process whereby the iron that would normally be in the ferric form is being chemically reduced to its more redox active ferrous form, iron 2+, in the presence of that amyloid when it's aggregating to form the plaques. And recently we were able to confirm that if you have ferrihydrite-like iron oxide cores in ferritin, if you incubate it at physiological pH, aggregating beta amyloid protein, there's a significant transformation of the iron oxide core to induce mixed valence, magnetite, and other chemically reduced iron phases too. So we've evidenced this mechanism in Alzheimer's disease tissue. We've also done this in APP-PS1 mouse models and in a series of in vitro studies to look at the impact of iron, or rather the impact of A-beta-42 aggregation on iron chemistry for iron solution, particulate iron, and so forth. And we've used soft X-ray approaches to revisit the amyloid plaque core work to confirm this in more detail. This is at the IO8 beamline at Diamond Light Source, going right down to tens of nanometer and building up stacks of images at different energies. And so this one here, this is, we get our plaque core image. And then if we move to 532 EV, we're getting signal from protein. We're getting contrast from that. We can get the carbonate signal, iron signal, and then look at a composite so we can see iron in the broader context of the plaque material. And so we can then look site-specific at the iron inclusions, if that's our focus, and then we can look at the spectra that we get from those individual sites to understand which mineral phases are present. So we're looking at very, very detailed studies within individual cores here. And in addition to doing the absorption spectroscopy to look at which phases were present, we were able to support that information with X-ray magnetic circular dichroism, which can also be done at the beamline with the sample in situ. So sort of moving between these different measurement methods whilst we've got a single sample set up on a single stage here. And in addition to that, we were also able to undertake titography and separate measurements, but also again with the synchrotron facilities to look at the morphology of inclusions. And the example shown here was really interesting to us because it had properties that were consistent with magnetite crystals that have been formed in living systems. And I think probably the most exciting thing that's come out of the work in the last few years is some work that we published in Science Advances last summer, led by James Everett, postdoc in our group, based at Keele University. And this discovery that we made to date, iron in humans is being found in positively charged forms, bound to various enzymes and proteins, and in iron oxide mineral forms. But here, with this synchrotron soft X-ray methods, we discovered a very small fraction, but very measurable and detectable all the same, of elemental metallic copper and accompanying ferromagnetic elemental iron in the human brain samples in these amyloid plaque cores. And these nanoscale pure metal deposits, they were found within the amyloid plaque cores from the Alzheimer's disease subjects, alongside other mineral forms such as magnetite. And we included site-specific X-ray magnetic circular dichroism measurements, as well as the absorption spectroscopy to confirm what we were observing. And we also documented the impact of the beam on the samples, a lot of measurements there to establish thresholds for beam damage. And we've described that process in detail in the publication because this was such a surprising observation that my first assumption was it must have been an artifact. So we checked very carefully before sharing these findings. And the discovery of metals in their elemental form in the brain raises some really interesting questions about their origins and the implications for disease pathogenesis. So this is something we're very keen to investigate further. And then when we're looking at these metal element inclusions, and I'm coming back to really looking at what techniques can do at this point, we can obtain really precise information about the inorganic components, but with these label-free approaches, it can be much more difficult to be sure of the location of the inorganic species relative to specific organic components in the tissue matrix, because this is all label-free. We're deliberately not staining because we don't want to introduce any contamination into the samples. So one of the recent things we discovered about the soft X-ray approaches is the extent to which we might be able to use features in the energy spectrum, the lower energies, to map for certain classes of organic material. And I think this is a lovely example from one of our postdoctoral researchers at University of Warwick, Jake Brooks. This is work he did as a major part of his PhD thesis. And he developed a way of imaging neuromelanin pigment in the human brain tissue sections. So this could be studied label-free in the context of these vulnerable dopaminergic neurons in substantia nigra that we know accumulate iron, and where neuromelanin is the primary iron-binding material in these cells. So here, by picking out this spectral component, tiny feature in the spectrum, looked at with neuromelanin that was produced. We made synthetic forms of it. We did it with and without iron binding. And we looked in Alzheimer's tissue, healthy controls, and brain iron accumulation cases at substantia nigra to show that we could map the neuromelanin in its own right. And here's an overlay up here where we've got the neuromelanin specific signal, we've got protein, and then a composite. And if you're interested in seeing how the iron ties in with that as well, Jake's paper was published in Angevanta Chemi back in 2020. So what I've been doing here is I've been showing you methods for looking in great detail at the properties of iron in tissue. But we also know from work in the field that regional patterns of iron distribution are reported to change as a function of disease. And so if iron status is useful to distinguish different forms of neurodegenerative disorders, we would like to be able to do this with magnetic resonance imaging, but we do have a fundamental challenge that when we're looking at MRI signal contrast in living individuals, we don't know for sure what the source of all that contrast is. We really do need supporting histology for that. And we wanted to look systematically at what happened to the relationship between MRI parameters and iron levels in tissues to see if we could do this spatially rather than relying on bulk quantitative analysis that's been done to date. And we wanted to do this in tissues that had not been stored in fixative to ensure that the metal ions wouldn't have had an opportunity to migrate within or leach from the tissues. So here we looked at the spatial correlation between relevant MRI parameters and iron distribution in human brain. So it's a very simple study concept. We've got a quantitative analysis of a block of tissue, and then after we've got our virtual slices in the tissue block. And then afterwards, we are physically sectioning it with a cryomicrotome and using X-ray fluorescence to map the iron distribution of the metals in the physical sections that have been aligned with those virtual MR slices where we've got the relaxation parameters. And we were able to confirm strong relationships between the MRI transverse relaxation R2 and the R2 star, which is influenced by local magnetic field inhomogeneities, and the iron distribution. And we found that working at 9.4 Tesla, which gave us a really nice opportunity to get microscopy resolution MRI, we found that the empirical relationship at the high field was consistent with the original field-dependent predictions from Weimersal and colleagues. And so in principle, this work can then be extended to test the relationship between the other elements present, because these X-ray fluorescence images that we've got over here, at the same time, if you remember back to that initial schematic for the X-ray fluorescence, we're actually getting the spectrum from all of the metals that are present. And so all this label-free information can be correlated to understand the influence of the different metals on the MR signal. And so we did some work towards this, and it's captured in Mary Finnegan's thesis, which is available from the Warwick Research Archive portal. And here Mary was starting to look at the relationships between the copper and iron and zinc in a couple of tissue regions. We were looking at substantia nigra and some of the brain regions as well, and the relationship with the MR parameters. And iron was dominating the signal, but I'm really highlighting this to show how we've got opportunities to explore the source of signal contrast in tissues to a much greater extent with this kind of imaging. So a couple of key point slides to summarise, and because this talk was about iron and neurodegenerative disorders and synchrotron approaches, I've actually split my concluding points over a couple of slides. So firstly, I'd say that important changes in iron metabolism in neurodegenerative disorders are being documented in the wider literature. This may aid diagnosis and therapeutic opportunities, so long as we've got sufficient insight into the mechanisms to make appropriate interventions. But we're not just looking at a change in distribution and concentration of metal ions. The chemical state and the mineral phase are also changing for certain metals, including iron, and that's the one that's most abundant and significant of the transition metals, and it significantly affects MR contrast. We also need to note that the altered patterns of transition metal distribution are used in diagnostics for certain disorders, but not yet for those that are the most prevalent, such as Alzheimer's and Parkinson's disease. So there is scope potentially to do more, even in these very complex disorders, where some of the changes are more subtle than in really rare disorders where there's a very clear and well-established signal. And then the last point for this slide is that the interdependence of metabolism for various metal elements and the implications for how we interpret signal in clinical data mustn't be overlooked. We've got this interplay going on. We've got iron dependent on copper homeostasis and vice versa. So all of these factors need to be taken into account. I don't think we can really afford to look at an element in isolation in this sense. And then the summary points where the synchrotron work is concerned. We've got really powerful approaches to allow us to address these complex analytical questions about iron and neurodegenerative disorders. So the synchrotron's a great opportunity in this regard. It's really important to be very innovative in the way that you approach these measurements. Traditional approaches don't always work. If you want to use the standard way recognised by others in the field, you may find you've really compromised your sample and it can't be used in that way. It's critical to have high integrity samples preserved appropriately for the property or properties of interest. So again, think outside the box. What is it that needs to be preserved for your measurement? And again, it may be different to what would be in routine practice. Really limited access to beam time. This is done through competitive access. You might get 1 week a year, 2 weeks a year at best. Measurements need to be really carefully integrated into a wider programme of work and planned. But we're finding the synchrotron measurements are really valuable when you're working at that interface between analytical metallomics in tissues and clinical imaging. So I'd just like to thank you all for listening and to acknowledge the many colleagues, including current and former members of our collaboration, who've made all this work possible, and a special thanks to the funders who've supported this work.
Thank you very much, Joanna. A very interesting lecture. And there's some questions in the question box. I put one too. I tried to answer them, if you can, inside the reply so everyone can see it. And you've kept to time perfectly. And now it only falls to me to introduce myself to give the next lecture. And I will share my slides, if that works. And here we go. And I presume you can all see this at the moment. Is that right? Someone say yes.
Yes.
Excellent. So it's a great pleasure to participate in this symposium. And what I'd like to share with you today is, if I can turn on my laser, here we go. I'd like to share with you a little bit of work that we've done in vivo in ischemic stroke and probing metals. And then I'd like to turn to brain endothelial cells as a cellular model to study the effects of hypoxia reoxygenation on the metal profile of those cell types, particularly the influence of physiological normoxia. But before I start, really, not all of these have funded all of the work, but one way or another, the British Heart Foundation, Heart Research, the Henry Smith Charity, and Evgen Pharma certainly have been involved with the research that I'm going to present today. And without the sustained support from Baker Ruskin, Agilent Technologies, and BMG LabTech, much of the work that I'll show you in cell culture under physiological normoxia would never have been possible. And you will hear, as I go through my lecture, certain aspects of their support. So in the first slide, what I'd like to do is introduce, as we well know, the blood-brain barrier. Here you can see the schematic taken from Joan Abbott's Nature Review Neuroscience. This is the endothelial barrier. It has tight junctions in health. Surrounding it are parasitic end feet or parasite end feet. We have the astrocyte that you see here, and then we have structures that lead out to the neurons and astrocytes, and we have an image of microglia. It is this barrier, the endothelial barrier or the blood-brain barrier, that preserves brain function in the environment, clearly regulating the flow of metabolites, and the junctions, as I'm indicating here from the schematic, are tight. However, in conditions such as stroke or ischemia reoxygenation, the reactive oxygen species generation will affect the integrity of the blood-brain barrier junctions, increase vasogenic edema, and allow permeability of blood-borne substrates into the brain parenchyma. Well, with the basis of that, I'm going to jump in my next slide to another schematic where I'm going to illustrate the consequence of a thrombus or a blockage of a cerebral artery. And you can see here that we have an infarct zone labeled here in the striatum. The blue is the infarct zone, and the region around it, this pinkish area, which I'm highlighting here in the larger inset, is called the penumbral zone. This is a zone that has not yet been damaged by the ischemic stroke or the blockage of blood flow, oxygen and glucose, and reoxygenation. And this is a region that we believe, or clinicians believe, can be rescued. And here's the intact zone. When we have a ischemic stroke, that is a transient stroke, on the y-axis here I'm illustrating the damage, and on the y-axis going downward, protection. Initially, you get a very rapid excitotoxicity. I'm not going to go into the individual components, followed over hours and days by inflammation and apoptosis, and then over weeks, scar formation is repair. If we look on the bottom part of this graph, we see that there's an anti-excitotoxicity component here. There's an anti-inflammation and anti-apoptotic component, and finally repair and regeneration. What I'd like to say is that this is not my own work. It's taken from Ulrich Dürnagel's Trends in Neuroscience and Endres in Cerebrovascular Disease in 2008. I'm interested in this early region with my group. In other words, how does the brain protect itself against radical damage? And one of the early studies by Peters, Dürnagel, and colleagues in the journal Cerebral Blood Flow, what they're plotting here is the chemiluminescent signal for a luminescent probe as an index of radical generation. Here's the middle cerebral artery occlusion in a rodent species. This is a rat where the skull has been opened and they're actually looking at the cortex, and they have injected a luminescent probe into the circulation. They do the stroke, so the blood flow is stopped, and here at this region, around 60 minutes after occlusion, they have reflow by removing a middle cerebral artery filament, i.e. the middle cerebral artery blockage is stopped, and you get an immediate radical, or should I say chemiluminescent signal indicative of radical generation. And it is this radical generation that is believed to cause changes in the patency of the blood-brain barrier that I illustrated in my first slide. Now, how would tissues that are exposed to radical damage defend themselves? And they do this by upregulating endogenous antioxidant defenses via this KEAP1, this is a chaperone protein as I'll show you, and this NRF2 transcription factor, which I'll walk you through the slide. Under normal non-oxidative condition, this transcription factor in the cytosol is tethered to KEAP1 and processed for ubiquitination via the 26S proteasome. When, however, cells and tissues experience oxidative, nitrosative, or electrophilic stress, the structure or the conformation between NRF2 and its binding partner KEAP1 is altered, so that this is now stabilizing NRF2, and this complex cannot be degraded via the 26S proteasome. Under these conditions, endogenous synthesis of NRF2 accumulates in the cytosol, and NRF2 can accumulate and bind to an ARE, abbreviated for antioxidant response element, together with a small MAF protein. Activation of the ARE in the promoter region of target genes leads to the upregulation of a number of NRF2 targets, heme oxygenase, a stress protein, NQO1, a phase 2 detoxifying enzyme, and GCLC, GCLM involved in glutathione biosynthesis. I can add to that what Poisson spoke about, the system XCT transporter, which exchanges intracellular glutamate for extracellular cysteine. The rest of this I'm not going to talk about. If you're interested in NRF2 redox signaling, there's a very nice detailed FRBM review from 2015. I'm not asking you to read 480 pages, but it has state-of-the-art reviews, and we're just producing another update of this in another FRBM special issue later this year. So basally, this transcription factor is ubiquitinated and deals with basal levels of radical stress, but under conditions of excess radical generation, oxidative distress, as coined by Helmut Sies, this leads to a response where this transcription factor upregulates protective genes. And we asked the question whether in ischemic stroke, activation of this pathway could be beneficial and protect both a rodent model from behavioral deficits. But before I do that, let me show you some data taken from Shah in Neuroscience. It's quite some years ago. This is the percent infarct from serial sections of the brain subjected in a wild-type mouse to ischemic stroke. You can see here the stroke hemisphere. And if the animal is globally knocked out for the transcription factor NRF2, you can see that in each serial section, there's a much greater deficit or greater infarct. In other words, NRF2 and its basal target gene expression is protecting the cerebral parenchyma and endothelium against subsequent ischemia reperfusion injury. So NRF2 is not just induced or activated in oxidative stress, it has a role to play under basal conditions. Now, the first thing that we wanted to do was ask the question, could we monitor in the intact mouse brain blood flow effectively during ischemia and upon reperfusion? And what we were very fortunate was from more instruments with the funding support from FGen Pharma to use transcranial laser speckle contrast imaging to monitor cortical blood flow, illustrated here in a hemisphere that's unaffected by a middle cerebral artery occlusion. And here you see the hemisphere where blood flow is markedly reduced because of the middle cerebral artery being blocked by a microfilament. What I'd like to show you in the next slide is the time course of this. We're here under baseline. You see, in this case, the stroke hemisphere will be on the right. This is ischemia. You can see the blood flow dropping dramatically here. And if we wait just a few seconds, we will remove the filament in the anesthetized mouse, and you will see a reflow or reflow that you can see labeled R, but you'll note that the reflow is not complete. And this has been referred to as the non-reflow phenomenon. And if we look here in the panel, this is still pictures taken through basal ischemia reperfusion. And we were very interested into whether we could use this compound. It is a dietary isothiocyanate called sulforaphane. It's found in cruciferous vegetables, for example, broccoli, and it's well known to activate NRF2. So here I'm showing you data from Antonio Cuadrado's lab in Madrid. They're looking at FSN in nanograms per gram tissue of brain after administering it. Here's the striatum and here's the midbrain. So you can see that within 15 minutes of an IP injection here of sulforaphane, we have maximum concentrations in the midbrain and striatum that then over time decrease. If I could take you to this instead, panel D, this is the NRF2 protein levels in time after injection of sulforaphane, and you can see that within 1 to 2 hours, there are significant increases in protein levels of NRF2 and its downstream targets. So this dietary isothiocyanate rapidly crosses the blood-brain barrier, activates the NRF2 defense pathway, and we will show you data now, hopefully that convince you it is protective against ischemic stroke. But before I do that, let me illustrate this little wild-type mouse here nibbling on broccoli. Mice don't like broccoli, and they certainly don't nibble it. So we gave the compound in a Nutella, small amount of Nutella, and mice love chocolate. We proved that chocolate didn't affect the weight gain or anything else in the mouse, but we were able to deliver sulforaphane very carefully to mice in the diet rather than intraperitoneally. The second take-home message for this slide is the following. Plasma sulforaphane levels after eating cruciferous vegetables in humans, for example, in normal dietary consumption, range perhaps maximally to 7 micromolar. Many of the studies conducted with cultured cells or animal models are using pharmacological or industrial doses of this dietary isothiocyanate, and it will have off-target effects. For those interested, have a look at this PNAS paper, but really it's these concentrations that are physiologically relevant. And what I'm showing you here in this still, if I look at panel E first, this is just taken from one of our wild-type animals here that you can see where the middle cerebral artery has been occluded. You see the lack of blood flow on the stroke hemisphere. If you treat animals for 3 days with dietary sulforaphane, you can see that you can markedly improve blood reflow here. And that's data shown up here. If we look at animals that are wild type, the black, if we give sulforaphane to a wild type animal, we can increase reflow or improve the non-reflow phenomenon. If we have animals that are globally knocked out for the transcription factor, we lose the ability of sulforaphane to rescue the deficit in blood flow upon reperfusion. So here we've shown that sulforaphane in the diet is able to improve blood flow in the brain, and any names that you see here refer to the PhD students or postdocs or colleagues that have done the actual work. Now here, what we want to look at in this slide is, does dietary sulforaphane in any way protect against motor and behavioral deficits in mice subjected to transient cerebral ischemia reperfusion? And here you see schematically a mouse walking across a ladder, and here in the bottom below, I'm showing you reperfusion time in days after the 1 hour occlusion. And these are the failed steps of the mouse. You'll see that animals treated with vehicle, these are the failed steps over time. And if we treat them with sulforaphane, we can improve this failure in behavior. Secondly, what we were able to show, if we do a sucrose preference test, that's offering mice sucrose or plain water, you'll see that in wild-type animals, the wild-type mice really prefer sugar water over plain water, but the NRF2 knockout mice are far less able to select sugar water versus non-sugar water. And then if we do the sucrose preference test, here against reperfusion time in days after a stroke, you see that over the first 3 days, we lose the capacity of these mice in both vehicle treated or sulforaphane treated to differentiate and select sucrose. But then as we go across with vehicle, the recovery is very slow, doesn't get back to control. But in animals that have been given sulforaphane, we can markedly improve that. So not only can we improve permeability of the blood-brain barrier, data I haven't shown you, we can improve the non-reflow phenomenon, and we can improve behavior and motor deficits using this in an in vivo model. Now, what happened is, as this came out and we were talking, the British Heart Foundation asked us to present to their lay audience our findings, which we did because they were funding our work. Finally, there was an interview that I gave to the Daily Express. I had no idea what they were going to write, but they wrote that we had identified a broccoli pill to save your life in stroke. And here you see breakthrough will prevent thousands of stroke deaths. I think they took some liberties to our animal model finding, but I show you this slide. This was the day Prince Harry and his wife met Meghan. and they were put to the bottom of the page. And this is the same day that Macron triumphed in the French election the first time. So this gave rise to our Evgen Pharma, but I thought it was a very interesting period to describe to you. Now, I'm going to, for the brevity of time, come now to oxidative stress and metals. Why were we interested, for example, initially with Wolfgang Marat, Thea, and other colleagues in zinc? Firstly, because activation of this pathway by oxidative stress can release free intracellular zinc. Secondly, zinc is involved in metallothionein. And finally, zinc can modulate the metal MTF1 transcription factor, and we believe it can modulate and affect NRF2. And as we're interested in antioxidant stress responses and metal binding and transporter genes, we began to look at zinc as a possible regulator or integrator of metallothionein and this redox sensitive transcription pathway NRF2. Here, I'm showing you one example done by Nathan Siddle, Alex Morell, and Thea Stewart. We're using laser ablation. I won't go over the methodology with you, and I'm showing you here an example of manganese, iron, copper, and zinc. The top two panels here are control animals, wild type or NRF2 knockout, and you can see that the metal profile is different. This is 1 hour after reperfusion. In other words, the middle cerebral artery was occluded for 1 hour, and the animal was then reperfused, or the blood was allowed to reperfuse the brain in wild type and knockout animals for 1 hour, and you see this profile. To our surprise, completely unexpectedly, 1 hour of occlusion with 72 hours reperfusion, this is the signal in the stroke hemisphere for manganese in both wild-type animals and NRF2 animals. Now, because I'm not a metallomics expert, this must mean that the manganese that we're seeing, this signal of total manganese content, must have come from the blood via the blood-brain barrier. Alternatively, if the blood-brain barrier was disrupted in ischemic stroke and reperfusion, this could reflect the influx of, for example, neutrophils into the brain carrying manganese SOD in its cargo. And just for those of you that are very familiar with metallomics, look at the signal here for manganese in the wild-type animal, look at the signal here for zinc, and then look at the NRF2. We have quite significant differences. What Nathan then set out to do is he looked at wild-type animals, control, 1 hour reperfusion and 72 hours reperfusion. And this is manganese 55 laser ablation shown here in a slightly different way. And this is the NRF2 knockout. If this is true, that this manganese is appearing from the bloodstream, crossing the blood-brain barrier as cargo, for example, in manganese SOD, an antioxidant enzyme, could we probe that in serial sections? And this is what Nathan did here. You see here a crescent violet stained brain. This is the stroke hemisphere, this white or lightened area, and this is the contralateral control hemisphere. This is a box drawn in the center of the infarct just here, and if we come across here and we stay in that section, immunohistochemically, from manganese SOD, you see a significant upregulation compared to the contralateral comparable region where manganese SOD levels are low. This is just GFAP and this is merged. So we're pursuing these experiments further to establish whether the increase in manganese SOD immunoreactivity correlates directly with the increased total manganese content in the ischemic hemisphere and whether this is driven by neutrophils. Let me see if I can. Now, let me just see here. A quick transition. Forgive me for doing this to you, but I'd like to do this. To look at the molecular mechanisms in more detail, we have to return to cell culture. And to do this, we wanted to work with brain endothelial cells. But before I do this, I need to really hammer home one concept. We breathe somewhere around 21 kilopascals oxygen, or 21% oxygen. And as it passes into the lungs, we know that in the airways, it's around 12 to 15 kilopascals, and the peribronchial regions around 5 to 6. If I take you across here through this schematic, this is air that we breathe, it moves in. This is the oxygen content in blood, starting in the air we breathe, not in blood obviously, and as we drop down this blue curve to a cell or a tissue, it ranges between 3 and 7%, depending on the organ you're in. In the brain, in different regions of the brain, and you can see more information in our physiological review from Tom Keeley that we published in 2019, oxygen levels can be as low as 0.5 kilopascals, and if we move up here near the peel surface, around 7 to 8 kilopascals. What I'm saying to you is, not once does a cell in culture ever encounter the standard cell culture oxygen levels that we use routinely, i.e. incubators gassed with room air, 18 kilopascals oxygen. The cells are exposed to hyperoxia. Now, how do we manipulate oxygen levels? Very simply, we use a oxygen-regulated workstation supplied by Baker Ruskin. Here you can see Tom Keeley and Salil Srivastava working in such a station. This is a microscope, a small Etaluma microscope. These are the cells where my cursor is signaling, and these cells are cultivated, treated, and extracted or imaged inside the workstation at either a defined physiological oxygen level of 5 kilopascals in this side of the workstation, or if you wanted to put them into hypoxia on this side of the workstation. The cells are never taken out of these workstations to a laminar flow hood, and thereby, by maintaining them long-term under defined oxygen, we can select a phenotype that is most accurately reflects what these cells or tissues would see in vivo. And this is data taken from Gabriella's, Matthew Smith, and others' work, where we're showing you that if cells are maintained in room air, standard cell culture incubators, HIF1 alpha, hypoxia-inducible factor, is not stabilized. If we put them in physiological normoxia, that's 5% oxygen, again, no stabilization of HIF if you leave them for 5 days, but at 1% oxygen in 3 independent cultures, as illustrated here, we clearly have stabilization of HIF and its downstream targets activated. Panel F is showing you catalase protein levels in air, standard cell culture, which are reduced if the cells are adapted to 5% oxygen. Similarly, total glutathione levels, a very key cellular antioxidant, shows you levels of around 30 in air. If we adapt the cells to 5% oxygen, this is lowered, and this is indicative, together with this, that the cells have experienced less oxidative stress because the oxygen level intracellularly, data I couldn't show you, instead of being 16 kilopascals or 60%, ranges between 3 and 5%, clearly having dramatic effects on the mitochondria, VR, and other cellular processes. This is recent work that has literally come out just a month or two ago, our collaboration with Emre Eroglu in Sabancı University in Turkey. What we've done here is taken HEC293 cells, transfected them stably with a genetically engineered NO probe, or we've taken endothelial cell line, stably transfected, and then we've looked at them simply as the basal signal in air, that's the 18 kilopascals for both cell types, or we've adapted the same cells to 5 kilopascals oxygen. And what we immediately note that if this probe sees NO, its fluorescence is quenched. So by moving cells to lower oxygen, we have less oxidative stress, more NO bioavailability, and that is why you can see here in the 18 kilopascals, the fluorescence, as you come from 18, this column here, to this column, you see less fluorescence because of more NO, and that's also shown here in these cells. If you took an external NO donor, this is NOx7, and you on the y-axis express it as 100% and a function of time. Cells that are sitting in air, standard culture, show this signal. Cells sitting in 5% oxygen, physiological normoxia, show this signal, both in HEC293 and endothelial cells. Finally, we're able to adapt our cells to different oxygen levels and use this instrument from BMG LabTech, which is an oxygen-controlled plate reader. And here we're looking at mean light units on the y-axis against time on the x-axis, and we're lowering the oxygen to hypoxia. So this is the hypoxic region, and here we're seeing reoxygenation. On reoxygenation, we get a respiratory burst. We're using a luminescent probe that is totally abolished in the presence of SOD, superoxide dismutase, or pegylated SOD, as you can see here. This panel here in C is the same experiment. We go to hypoxia. This is the signal in cells that have been treated with vehicle, and the green is cells pre-treated with sulforaphane. The same agent that I showed you earlier in vivo protects the brain against reperfusion-induced oxidative stress. And you can see that we can markedly lower the redox signal by pre-treating with sulforaphane, and data that I don't have time to show you, we know that this is because sulforaphane pre-treatment has upregulated NRF2 defense pathways. And finally, I'd like to end with this set of data. These are hypoxia reoxygenation experiments using ICP-MS to analyze the metal content in mouse brain endothelial cells. Shown here, if we just, let's take this panel here, which is zinc 66, nanograms per microgram protein. You can see here under basal conditions, irrespective of the oxygen they're adapted to, we have a similar level. As soon as we have hypoxia or hypoxia reoxygenation, we are doubling effectively the zinc content, we're doubling the manganese 55 content, and we're doubling, if you see here, magnesium 24. This suggests that the hypoxia reoxygenation protocol in these cell types, in this cell type rather, is increasing the delivery of metal from the medium into the cell, and we're exploring which transporters are involved. And finally, work taken here below is from Fan Yang, my PhD student, is looking at NRF2 nuclear to cytoplasmic ratio. These are cells under vehicle condition in physiological normoxia, stimulated with sulforaphane. We activate NRF2 nuclear levels. This is giving external zinc. This is the interplay between zinc and NRF2, and this is in the presence of TPEN. So this is all I was going to share with you on this, but I want to leave you with two review articles that please have a look at. One is our physiological review articles, and this is a recent Nature, it shouldn't say Nature, in February 2022. Adapting cells to physiological oxygen. These are cells that are endothelial cells that have been challenged with ionomycin, a calcium ionophore, big damage to annexin 5 staining. Cells pre-adapted to physiological normoxia show virtually no damage, and this has major implication for calcium overload, both in the brain and in the heart in ischemia reoxygenation injury. And what I'd like to leave you with is a plea for a paradigm shift to please adapt cells in culture to oxygen levels they see in vivo, so that we can inform both disease pathology and the design of novel therapeutics. My group, or the group of Richard, myself, and others, there's Richard, myself, Paul Fraser, Richard Naftalin, a few others. There's Matthew Smith. This is Tom Keeley, Salil, Gabi Warpsinski, Will Pickles, Ron Jacob worked in the brain, Fan Yang works with coronary artery, smooth muscle cells, Steve Williams, Alessio, and my friends here in the stroke, Nathan, Paul, and Keith were critical. I would never have done any of this if I'd not met Tetsuro Ishii in Japan many, many years ago. And with that, I'd like to say thank you. Okay, so now I get rid of that. I unshare. I apologize. I think I went over. How do I get back into this? Right, I'm stopping to share. but you can hear me, right, everybody, at the moment? Somebody?
We can hear you.
Thank you very much. And it is now, you have to punish me for going over, Costantino. I'm really sorry, but it's my greatest pleasure, personal pleasure, and on behalf of the London Metallomics facility, to have you to give the keynote lecture for this second annual symposium. And rather than read all your accolades, Costantino, we've known each other for some time. I would like to just say, he's in the top 1% of the most cited authors in the field of neuroscience and behavioral sciences. And I think your lecture is directly pertinent to me, so I've been extremely selfish, but I think my colleagues will also appreciate the context of your talk. When you talk about innate and adaptive immunity in stroke and dementia, challenges and opportunities. Costantino, thank you very much for joining us.
Thank you, Giovanni.
Are you going to share your slides that you have?
Yes, I
Good.
will.
Thank you.
He says, I cannot share because I'm not a presenter.
Richard, can you try to help, or? Just a second. We will sort it. Richard, are you live, Xiao?
I think Costantino is a presenter now. Can you try again?
Yep. It says open share tray. Should I just go ahead and share and see what happens?
Yeah. Go and share, and then click on the desktop.
Do you see anything?
No, not right now.
Yeah.
Okay.
If I do, it says open share tray.
In the button at the top, when you see in Teams, you see a little arrow just where it says don't go leave. Yeah. But if you press that arrow,
I did that. When I click on it, it says open share tray.
It doesn't show you screen?
No. I still see yours. Yeah. I still see your image.
Okay, I'm just going to get rid of that. Okay. Richard, what can we do?
Hello.
Or Brian.
The alternative is if I share the presentation and Costantino, if you speak to it.
But then he can't use his cursor or anything, Richard.
Hello, Costantino. Are you using a Mac, not a PC?
Yeah, Mac, but we have tried that before.
Yeah. And I used the Mac just now.
Yeah, it went all right when we did the test.
We tried that in the couple of days ago.
Yeah, you want to do what we did last week. I think it was try and log off and come back in again.
Okay.
Because it worked last time. Try that.
Thank you, everybody, for hanging in.
If that doesn't work, I've got your slides ready to share.
Thanks, Richard. Just give them a few minutes to come back.
Okay, I'm admitting. You're back? Yep, he's back twice.
He's back now?
Yep.
Still on mute.
Yep, they're here, your slides. If you just press the big, can you hear us, Costantino? Yep, we can see your slides, but we can't hear you.
Can you unmute, please?
You have to, there we go.
Giovanni, thank you very much for your short introduction, because this way I can get through my slides. It will not be too long. So we're going to kind of take on, wherever we left off in terms of the stroke and dementia, but we will focus on the role of immune factors in these two major neurological diseases. And I'm going to tell you a little bit about stroke, perhaps taking wherever Giovanni left it. And then introduce the concept of dementia and focus on the vascular contribution to dementia, and with respect to risk factors as well as, Alzheimer pathology. So let's take a real, this is not a mouse, it's a human that has a stroke, a middle cerebral artery occlusion, which is the most common form of stroke.
We don't see your slides at the moment.
No?
No, they were there and then gone. Let's see, I'm sorry.
How about now?
No, we just had them, but then when you started talking, they went away.
Oh boy.
Hello, Constantina, what do you see now on your computer?
I see just Giovanni's image.
Can you not see your some thumbnails of your?
I see my thumbnails in the bottom right.
Yeah. Your slide?
Do you see my slides or no?
No.
I lost that little tray that says that I can do the slides. Oh, here we go. Now it's there again.
There they are.
Okay, yeah, that's great.
Okay, let's do this now.
Try again.
There.
Okay, great. So, all right. So this is a real patient. Can you hear me and see everything?
Yes.
Okay, so with a middle cerebral artery occlusion, goes to the hospital. In any large, city hospital, what they would do is do a measurement of cerebral blood flow. The poor man version is called perfusion weighted imaging. It's not quantitative, but it gives you an image of what's going on with the blood flow in the brain. This is the front of the brain, back of the brain. As you can see, there is an area of flow which is very, very low. This is what Giovanni called the ischemic core. And then there is a surrounding area with larger, in which blood flow is a little bit higher, but not quite as normal on the other side, which is called so-called ischemic penumbra. Now, if you do diffusion weighted imaging, which is an index of early damage, you will realize that in the area where the blood flow is very, very low, there is already early damage. This is a couple of hours after the stroke has occurred. So this is there is no hope for this region. Now, if you subtract the area of damage from the area of reduced blood flow, you have an area which the radiologists call the mismatch, which indicates the brain which may be at risk for stroke because it's got a blood flow reduction, but not quite to the level that causes the brain to die. Now, if you wait 12 to 24 hours and do nothing, what happens is this area of the diffusion weighted imaging signal expands to encompass the entire area of the flow reduction. In other words, the brain at risk became dead brain. And the cause of this transition is the so-called ischemic cascade that Giovanni also alluded to, which is a chain of events which is initiated by the energy failure caused by the arterial occlusion and then takes on a life of its own. And it goes on and on and on, destroying the brain and then setting the stage for brain repair at the same time. So that's the so-called ischemic cascade. Inflammation and immunity play a major role in all these phases of this process. And we're going to be talking about this. Now, when you have an occlusion, arterial occlusion, you got things happening in the brain with respect to immune cells, but you also have things happening in the periphery. And so before we go on and describe what goes on in the brain, let's talk about the periphery. So ischemic stroke is like hitting the brain with a hammer. And that leads to a massive activation of the autonomic nervous system through the hypothalamic hormones, as well as autonomic nerves, originating from the brain and through the spinal cord and all the sympathetic and parasympathetic distributing itself throughout the whole body. So this leads to a massive autonomic storm in the periphery, as well as together with the danger-associated molecular patterns, which are molecules released by the brain, which is dying into the circulation, leads to a massive activation of all the immune organs. And this leads to an immune activation. So essentially, there is an increased cytokines, there is a proliferation of white cells, and so on. However, this immune activation is followed within days to a immunodepression. And the reason why I'm mentioning this is because the major cause of death in stroke are infections that occur after the stroke has taken place, particularly pneumonias and UTIs, urinary tract infection. So this kind of peripheral storm that occurs is very, very important to the survival of the patients. And so which I also receive a lot of attention in clinical trials, because you would want to try to kind of minimize this source of morbidity and mortality that is not related to the brain itself, but to the impact of the brain on the systemic immunity. Now let's look at immune factors within the brain itself. So when there is ischemia, reperfusion, the hypoxia, the free radicals which are generated lead to a adhesion of circulating leukocytes to the cerebral endothelium via the expression of adhesion molecules, some of which are already present within the endothelium, like the P-selectin, that are simply translocated to the surface of endothelial cells, leading to the attachment of these neutrophils, which normally would just fly by, but all of a sudden they stick to the endothelium in the areas of injury, and leading to an opening of the blood-brain barrier through production of cytokines, oxidative stress, and so on, which then leads to the extravasation through paravascular pathways, as well as other pathways that we will see, from the vasculature into the brain. So these cells are really white cells, neutrophils, monocytes, and also T cells, as we will see, that come into the brain. They are activated. They have a lot of nitric oxide production through the enzyme inducible nitric oxide synthase. They make industrial amounts of radicals, because that's what these cells do, and they produce cytokines contributing to the damage. Here we have a cell that is exiting a venule in the act of exiting. That it shows you kind of how dramatic this process can be. Now, at the same time, there are things happening within the brain and in the meninges of all places. So within the brain, the ischemia, the reduction in blood in oxygen and caused by the arterial occlusion, leads to early damage to the neurons and glia, which are very, very sensitive to hypoxia. At the same time, the invading neutrophils release metalloproteases, radicals, as we said, nets, and so on, leading to the contributing to the damage to the cells. So this generates these danger-associated molecular patterns, which are fragment of membrane, DNA, phospholipids linked to proteins and so on, which are able to activate innate immunity-driven cells, like, for example, microglial cells and macrophages within the brain, again, leading to cytokine production, and also amplifying this damage, which initially was only an ischemic, hypoxic damage in nature, but now it becomes much more pervasive through this immune compromisation. At the same time, there is a exchange between the meninges, which have emerged now as a major reservoir of immune cells within the brain. And just a couple of years ago, it was first described that the skull bone marrow, so the bone marrow of the skull, within the skull, is the major source of some of the neutrophils that get into the meninges and then end up getting into the brain. So as you can see, within the head itself, there is almost a self-contained machinery to kind of contribute to ischemic brain injury. And as the bone marrow in the skull contributes more than the bone marrow of the tibia or and so on, or other long bones, to the accumulation of these immune cells in the brain. So this is really very, very much bad news for the brain. And here you can see a patient who died 24 hours after stroke, and you can see this kind of cuffs of inflammatory cells surrounding an arteriole, as well as the clotting within the brain, the vasculature, and then invading the brain, many of these cells express inducible nitric oxide synthase, a major source of NO, as well as nitrotyrosine, which is the chemical that comes from the reaction of nitric oxide with peroxynitride, and is thought to kind of expand the damage because peroxynitride is more diffusible than nitric oxide that gets quickly captured by superoxide and so on. So you can see this oxidative, stress damage affecting the brain. Now, how do these innate cells within the brain, like the microglia and the monocytes and the macrophages and so on, they lead to cell death by producing free radicals and inflammatory cytokines via activation of innate immunity receptors, some of which are the toll-like receptors, some called scavenger receptors like CD36, that leads to activation on FKB, inflammasome activation, and production of cytokines that lead to pyroptosis, as well as production of large amounts of free radicals that lead to oxidative stress. So these are kind of the effectors of the damage eventually caused by this innate immune activation. But what about T cells? T cells can be both innate or can be also in response to adaptive immunity. And in ischemic injury, T cells play also a very important role. In particular, there is a balance between the negative effect of gamma delta T cells occurring in the acute phase of cerebral ischemia, so in the first hours, and perhaps a couple of days after ischemic brain injury, through the production of IL-17, which is a very toxic cytokine. At the same time, if you wait, a week or so, you have another T cell called the regulatory T cells, or Tregs, that are protective via the production of IL-10, which is interleukin 10, which is a protective cytokine. So the outcome of the brain, once the ischemic injury is kind of done for, it depends really on the balance between these two population of T cells that determine how the ischemia will lead, how this kind of post-ischemic phase, early post-ischemic phase, will contribute to this evolution of the damage that occurs, over the next several days. Now, a critical regulator of T cells in the body is the microbiota. So the microbiota, which is the bacteria that are located in all our exposed mucosal surfaces. Now, of these, the gut microbiota is really the major player because in the majority of lymphocytes within the body are within the gut. So the microbiota is continuous exchange with the gut lymphocytes and play a major role in all the diseases in which adaptive immunity mediated by T cells plays a role, including brain diseases like multiple sclerosis. So considering the relationship between the gut microbiota and the T cells, we, a few years ago, were wondering whether the microbiota may have some kind of effect also on this T cell trafficking and protection versus destruction that occurs in the post-ischemic brain. So we include some of my colleagues, Josef Anrader and two postdocs, which are now have their own lab in Europe. We approach this issue. So now, how do you study the role of the microbiota in brain disease and stroke? Well, you can do it two ways. One way is to use germ-free mice. However, germ-free mice are not all that great because they have a blood-brain barrier alteration to start with. So you need the normal microbiota to develop a blood-brain barrier, right? So I don't think in stroke you would, as Giovanni mentioned, the blood-brain barrier has a major role. So you don't want to use the mouse, which already has a problem with the blood-brain barrier. The other way is to use antibiotics to kind of induce a dysbiosis, what's called an alteration of the normal microbiota. So the problem with antibiotics is that they can be profoundly neuroprotective. So you're not going to be able to tell what you see as a result, whether it's due to the antibiotics or something else. So to overcome this problem, we used a special mouse which has been on antibiotics for generations. And this mouse has developed a gut-resistant flora to antibiotics, particularly amoxicillin and clavulanic. So what we did, we co-housed regular black 6 mice with this special kind of mice, as a seeder, as a provider of the microbiota. The mice are coprophagic, and within a few days, they collect the feces of the other animals, and they assume the microbiota of the host that is put into the cage. So then both groups of mice, these mice with resistant flora, acquired from the seeders, as well as the mice with the sensitive flora, which were like regular mice, they were put on antibiotics, and as a result, the resistant mice can maintain the microbiota they had because they already were accustomed to the antibiotics. But the sensitive mice developed dysbiosis because they'd never seen the antibiotics before. All right. So what we did then, we gave antibiotics to the mice. Then after 2 weeks, we collected the stool for a 16S assessment of the microbiota. And then we produced a middle cerebral artery occlusion using the filament method that Giovanni described before. And then we assess behavior and in quantification of brain damage later on, between 3 and 7 days. And the results are shown here. So here you can see that the normal flora of the mouse, which has an antibiotic resistant flora, and take my word for it, or look at the paper, and this flora is exactly the same as the one we see in a normal wild type mouse, all right? But is in an animal which is on antibiotics. The guys instead who had a sensitive flora because they'd never seen the antibiotic before had a profound alteration of their microbiota, resulting in an expansion of the proteobacteria and a reduction of firmicutes. So the diversity of the bacteria was extremely suppressed, which is a sign of dysbiosis. And then we looked at the stroke volume in these mice, and what we found that these guys had a massively reduction in stroke volume. So the injury shown here by the white area in these crazy virus stained sections was much reduced. And also there was an improvement of the sensory motor function, indicating that this reduction of pathological damage was also reflected in an improvement in function. Now, the next question is, how does this happen? How can you have something happening in the gut kind of had such a profound effect on the brain? So we started this kind of long trip through the microbiota, the gut, brain axis, and so on. And we started with assessing the inflammatory cells within the brain on the assumption that this had something to do with immunity because of the T cell business and the gut. So we did flow cytometry looking at all the cells, cell types, immune cells in the brain. And what we found that there was a tremendous reduction in inflammatory cells, particularly in neutrophils. And these neutrophils are the ones that mediate the acute damage after stroke, in this post-ischemic period. And so this reduction of kind of the infiltration of neutrophils was reflected with a suppression of all the chemokines that led the neutrophil into the brain after stroke, CXCL1, CXCL2. So it looks like, with this microbiota in the gut, we were kind of reducing the ability of the neutrophils to get into the brain because the cytokines that drive their translocation into the brain were not being produced in the brain. Now, to make a very long story short, what we found was the following: that when you have a stroke, you have a disactivation of systemic immunity that leads to a alteration of the balance between these two major T cells that affect the outcome of cerebral ischemia. There is an increase in gamma delta T cells, which make IL-17, which is bad for you, and a suppression of regulatory T cell, which make IL-10, which is good for you. All right. By this, then these cells end up in the meninges of all places. I told you that the meninges, are now at the forefront of stroke pathobiology. They end up being in the meninges, and they produce cytokine that then go into the brain, and they favor the infiltration of white cells, resulting in an enlargement of this kind of pro-inflammatory component of ischemic brain injury. Now, what happens with the microbiota is that the microbiota modulates this alteration such that there is a suppression of gamma delta T cells and an upregulation of regulatory T cells. So the balance is shifted completely to the other, towards a protective T cell phenotype. As a result of that, there is a suppression of meningeal gamma delta T cells, a reduced infiltration of neutrophils, and reduced lymph volume. So the data really highlights, how critical is the microbiota in the kind of determining, the overall outcome of ischemic brain injury. Now let's move on. And I told you what's going on when the brain gets hit with a vascular occlusion, which leads to energy failure, ischemic brain injury, and cell death, acute cell death that occurs when the blood flow is completely occluded and doesn't get to the brain at all. However, there are other conditions where there is a more subtle alteration of the delivery of blood flow, which means oxygen and glucose to the brain. And this is caused by vascular dysregulation that lead to a kind of mismatch between what the brain needs in terms of oxygen and glucose to satisfy its energy needs and the delivery of blood flow. So when there is this kind of neurovascular dysfunction in which the vessels do not really follow the request of the neurons and glia for blood flow, then in this condition, often there is cognitive impairment. So now we're going to talk about this other situation which is not so extreme, but nevertheless is also critical because it leads to cognitive impairment, another major cause of morbidity and mortality. So let me tell you a little bit about the concept of Alzheimer's disease and concept of dementia over the past, 100 years or so. At the time of Alzheimer, we're talking about early 1900s, the major cause of dementia was thought to be hardening of the arteries. However, after Alzheimer described this condition, we characterized by accumulation of plaques and tangles within the brain of these patients. The field, particularly in the 1990s, where there was a lot of molecular studies, unveiling the molecular pathology of Alzheimer's disease, led to completely forgetting the role of the hardening of the arteries. And so everybody really, the field was captured by the concept of Alzheimer's disease. Why? Because they were starting to make headway into the molecular pathology. So, for example, the identity of the peptide that accumulates in the plaques was described to be beta amyloid. All right. The genes causing familial Alzheimer's disease were starting to be discovered, and the concept of presenile dementia and the dementia that runs in the family because of mutation of presenilin genes, mutation of the amyloid precursor protein, and so on. So there was a tremendous optimism in terms that essentially dementia of the age could be really solved within a few years, and obviously this didn't happen. At the same time, however, there was an increasing literature suggesting that Alzheimer's disease, meaning plaques and tangles, were not the whole story. But in the dementia that occurs in the aged population, vascular risk factors also played a major role. And in fact, adding hypertension, diabetes, or hypercholesterolemia would double your risk to get Alzheimer's disease. Okay, so suggesting there was a commonality of mechanistic processes. In addition, the NUN study, a landmark study, clinical pathological study, revealed that when there were small ischemic lesions in the basal ganglia, like this kind of what we call ditzels in the basal ganglia seen on an MRI, which have no impact on cognition whatsoever, when these little lesions were associated with little bit of Alzheimer's pathology, which means plaques and tangles, the dementia that the patient experienced was much more exaggerated. So in other words, having these ischemic lesions, aggravated the cognitive impact of Alzheimer's pathology. So this suggested to some that there may be a synergistic effect between ischemic brain damage, even minor, as well with the pathology associated with Alzheimer. And the two of them together would lead to a synergistic amplification of the cognitive impact. Now, this idea is gotten much more accepted when we recently, when the comparison was made between the clinical diagnosis of Alzheimer. So this is the diagnosis made in the doctor's office and the pathological diagnosis of Alzheimer made at the autopsy table. As you can see, clinically, most patients, 70% of the patients with age-related dementia are said to have Alzheimer. However, at the pathological table, only 24% of them will have plaques and tangles. The majority of them will have a combination of vascular and Alzheimer's disease pathology. So mixed dementia is really the most common cause of dementia in the age. It's not Alzheimer, it's not vascular dementia, but it's a combination of the two. Now, ironically, the latest kind of development in the molecular pathology of Alzheimer's disease came a couple of years ago in a proteomic study in which patients with the minimal cognitive impairment or Alzheimer's disease, where the brain, from particularly the prefrontal cortex, was removed and analyzed for protein content and identification, and correlated with intracranial atherosclerosis, which is really the hardening of the artery of Alzheimer. And what they found was that the hardening of the arteries of Alzheimer that Alzheimer described 100 years before was an independent pathogenic factors in Alzheimer's disease. And some of the mechanism as derived by ontology analysis from the proteins were really pretty much overlapping with those of tau. So here we have atherosclerosis in the brain kind of contributing to cognitive impairment through mechanisms which are very much identical to those in which classical neurodegenerative pathology causes, like, for example, tau. Right? So this really brought back to the forefront the importance of vascular factors in the pathology of Alzheimer's disease. Now, this should not surprise anybody because the brain is uniquely dependent on a good delivery of blood flow in order to survive. So there are mechanisms like functional hyperemia, which is the ability of neurons to increase blood flow when they need more, when they are consuming more oxygen and glucose. For example, if you move your contralateral hand, in your brain, you will have an increase in blood flow in the supramentary motor area and here, and in the somatosensory and motor cortex, as was described, years ago by Ingvar and Lassen. And then there is also autoregulation, which is the ability of the brain to keep blood flow more or less constant despite changes in arterial pressure. Why do we need autoregulation? Because blood pressure fluctuates by 20, 30, 40 millimeters of mercury, depending on what you're doing. And if there was no autoregulation, blood flow to the brain will go up and down constantly, which is not going to be very good for the brain. Furthermore, the endothelial cells of the brain are also very important for regulation of blood flow. They release vasoactive factors like nitric oxide, which was going to come up later in the talk, and they also are at the site of the blood-brain barrier. Now, so these factors, maintain the homeostasis of the cerebral microenvironment by feeding the brain and doing many other things to the brain. However, in conditions like Alzheimer pathology, or for example, in vascular risk factors like dietary salt, these factors that regulate the cerebral circulation are altered, and it leads to an insufficient delivery of blood to the brain that results in cognitive impairment. And we're going to examine now some of the contribution of these. Let's start with Alzheimer pathology. So the fact that vascular risk factors and vascular pathology contribute so much to Alzheimer's disease raised the possibility that A-beta and tau, in addition to producing neuronal dysfunction directly, which is very well established and nobody's arguing with that, may also alter the cerebral circulation, thereby constituting another hit on the neurons. So not only they are directly affected by disrupting synaptic function through the neurophysiological effect of A-beta and tau, but there could be also another negative effect of lack of sufficient blood to the brain. So a number of years ago, we had the opportunity to study this phenomenon and whether A beta plays a role. So I'm going to briefly tell you how we study the regulation of cerebral circulation in a mouse. All right. So what we do, we open a cranial window. Essentially, we drill a hole on the head of the mouse, and we expose the cortex, and then we record blood flow using a variety of methods at the site. To activate the brain in order to study neurovascular coupling, we use the whisker barrel cortex model. So each whisker of the face of the mouse corresponds to about 2,000 to 3,000 neurons in the so-called whisker barrel cortex, which is a region of the somatosensory cortex. So if you stimulate the whiskers mechanically, you can increase blood flow very neatly into a sub-region of the cortex corresponding to a particular whisker, which is independent of blood pressure, blood gases, and so on. Now, because then the cortex is exposed, because there is a hole on the head, you can, if you topically applied acetylcholine on the brain, you can study the ability of the endothelial cells to increase blood flow. It's called endothelium-dependent vasodilation. So with this method here, you can really look at everything that has to do with the circulation of the brain. So we were, a few years ago, many years ago actually, we had the opportunity to study one of the mouse models of amyloid accumulation, which had just been introduced, the TG2576 mouse, studied at a very, very early age, 3 months, when there is no Alzheimer pathology yet, so there is no plaques in the brain, and the mouse is cognitively normal, all right? So there is nothing else going on. So we looked at vascular regulation. We found that there was a profound attenuation of functional hyperemia, endothelial function, and there was no autoregulation at all. So essentially, all the fundamental mechanisms that regulate the cerebral circulation were completely shut, as we showed. So based on this data, and based on the fact that this alteration precede the accumulation of amyloid and precede the cognitive impairment, we propose that perhaps this vascular dysfunction could be one of the earliest manifestations of Alzheimer's pathology in this, what we used to be called the prodromal phase of the disease, or latent phase of the disease, when there is no really any cognitive impairment, but there is still something going on because A beta is starting already to accumulate. We know now that A beta accumulates 20 years, start to accumulate 20 years before people forget their first words, all right? So there is a lot of things going on in this latent phase. So we thought that perhaps the vascular alteration was occurring early on, such that, would precede the other aspects of the disease. And we recently a study in the so-called ADNI cohort, where all the biomarkers of Alzheimer's disease were correlated with the progression of the disease from early minimal cognitive impairment, late minimal cognitive impairment to full-blown dementia. Vascular dysfunction was found to be the first alteration that can be detected, confirming what we saw, years earlier in the mouse model. So vascular changes are very, very significant in the pathobiology of Alzheimer's disease. Now, what mediates these changes in the vasculatures? Well, it's our enemy, which is the oxidative stress. And today you've heard a lot about oxidative stress. So as you can see, this is a resistance arteriole, a PL arteriole in the brain. You can see there's massive oxidative stress, as shown here, by peroxynitrile accumulation. The source of the oxidative stress is an enzyme called an ADPH oxidase that is initially described in neutrophils, but now has been found in many, many cells. And this enzyme is activated in the brain by an innate immunity receptor called CD36, which is able to bind the beta, activate an ADPH oxidase, and leading to oxidative stress. At the same time, this enzyme or this CD36 also activates an FKB, leading to inflammation. So inflammation and oxidative stress are really the major factors in this vascular component of amyloid dysfunction. So the data then suggests that, A beta, which is produced by the brain and is cleared, some of it, through the vasculature, okay, reaches the blood vessels of the brain, engages a cell on the vessel wall called a perivascular macrophage, activating CD36, which is this innate immunity receptors. The macrophages are loaded with NOX2, leading to massive production of reactive oxygen species that produce all the endothelial dysfunction, functional hyperemia, altered regulation, blood-brain barrier, and so on, that we have seen in both in the mouse model and in patients with Alzheimer's disease. So these perivascular macrophages can be seen here, plastered against, penetrating arterioles in the brain. They are really a major source of oxidative stress at the vascular level because they're just, stuck on the surface of the blood vessels in this so-called perivascular space that exists in the brain. Now I'm going to tell you about another form of kind of cognitive impairment, which is associated with the excessive salt intake. High dietary salt has been linked to stroke and dementia for many, many years. So in this study, you can see the excretion of sodium in the urine, which is the best way to tell how much sodium you're taking through that, correlated with the mini-mental state, which is a way of measuring cognitive ability. Normal cognition is thought to be 30. As you can see, the more salt in people assume dietary salt, the worse is their cognitive function. The reason of this is have remained unknown for a while. So a number of years ago, one of my colleagues, Dr. Furaco, undertook the task of figuring out what was going on. So we developed a mouse model of cognitive impairment caused by salt. And what we did, we put mice, black 6 mice, on a high salt diet, very, very high level of salt. And we chose a mouse strain in which there is no increase in blood pressure when you get salt. So in about 30% of people, when you give them a salt load, they increase their blood pressure. It's called salt-sensitive hypertension. In the mouse, it's the same, but we chose a strain in which there was no hypertension. Why? Because hypertension is a major confounder because it also affects blood flow to the brain. So we didn't want anything to do with that blood pressure. So what we found was that this high salt given for weeks led to reduction in resting blood flow to the brain by about 25–30% in the cortex and hippocampus, altered endothelial function, attenuating the response to acetylcholine, so the endothelium is unable to increase flow anymore, but did not affect functional hyperemia. So unlike the Alzheimer model, right, where everything is gone, here instead we have a selective problem with the endothelium. And when I saw this, I was not really convinced that this was going to lead to major phenotype in a mouse, but I was completely wrong because not only the mouse were cognitively impaired in a major way, so they couldn't tell, for example, a novel object, a new object from one that they were familiar with, which is the mice do very, very well. But they also were unable to build a nest, which is activity of daily living for a mouse. So a mouse cannot build a nest is really very much screwed up, mentally. So this very puny, smallish alteration in blood flow to the brain was leading to this massive phenotype, a cognitive phenotype. And so we were very, very surprised and eager to find out what went on. It wasn't easy, but eventually we came up with, we kind of had an hypothesis and some data to support it. So what happened is when you feed high salt diet to a mouse, as in a human as well as been shown, you have an expansion in the gut of a special group of lymphocytes called Th17 cells. So these cells, they make large amounts of IL-17. I told you before is a cytokine that is not very good for the brain. So, and you can see this increase in IL-17 was shown by flow cytometry to be mediated by Th17 cells. And these cells make so much of it in the gut that it spills over into the circulation, leading to an increase in circulating IL-17 in the blood. So this IL-17 then interacts with IL-17 receptors on endothelial cells, activate Rho kinase, leads to a post-translational modification of endothelial nitric oxide synthase, or eNOS, that leads to suppression of enzyme activity, is an inhibitory phosphorylation, that leads to an endothelial nitric oxide deficit and cognitive impairment. Now the question was then, at the end of this, this first paper, what is the mechanism whereby this endothelial node deficit leads to cognitive impairment? Is because there is a reduction in blood flow to the brain, or is because of something else? Personally, I never thought that the flow was going to be pretty much playing a role, because the lesion, the alteration was relatively minimal. And then there was something that struck our hypothesis. It was shown by Dr. Katusik laboratory a few years earlier that the cerebral endothelium was able to suppress the phosphorylation of tau. Now, tau is one of the major factors in neurodegeneration. It's involved in Alzheimer's disease. It's involved in other diseases, like for example, frontotemporal dementia, in which accumulates and is very bad news, all right? So, and this study showed that nitric oxide from the endothelium, surprisingly, was able to kind of shut off tau phosphorylation in neurons by acting on CDK5 activity, which is the major enzyme, one of the major enzymes phosphorylating tau. And so based on this, we developed the hypothesis that perhaps what's going on here is that nitric oxide from the endothelium nitrosylate calpain, which is the enzyme required for CDK5 activity. And so by keeping calpain at bay, also CDK5 activity would be kept inhibited, and there was no excessive tau phosphorylation. In the presence of endothelial dysfunction, the reduction of nitric oxide, which I showed you occurs, will lead to denitrosylation of calpain, unleashing CDK5 activity, which then would promote tau phosphorylation, misfolding, aggregation, and brain dysfunction. So to test this hypothesis, we measured tau in the brain of mice on a high salt diet. So this is the cortex hippocampus, normal diet, high salt diet. As you can see, there is a massive increased phosphorylation of tau at all the major phosphorylation sites which have been implicated in cognitive impairment in Alzheimer's or FTD or whatever. We saw accumulation of tau, but no tangles, all right? So the tau was still in a low aggregated state, was not quite, in a tangle form. But we saw that there was a suppression of calpain nitrosylation, an increase in calpain activity, and an increase in CDK5 activity, which was very much along our hypothesis that there was this signaling pathway was involved. However, the fact that tau is phosphorylated in the brain doesn't mean that it's causing the problem. And why is that? Because tau can get phosphorylated in the brain by many things, some of which are very protective. For example, hypothermia, which is the most protective cerebral intervention that you can do, induces massive tau phosphorylation. Hibernating animals, have huge amounts of tau phosphorylation in their brain, and they wake up, they're fine, all right? So the fact that there was tau around, phosphorylated, doesn't mean that that the brain is not working. So to prove that really tau was causing the problem, we used antibodies directed against tau. And these are the same antibodies used in clinical trials now to counteract the deleterious effect of tau. And what we found was that when the mice were treated with antibodies, they were cognitively normal, despite being on a high salt diet. So essentially, by suppressing, the effect of tau, we were able to normalize cognition. So the next question is, what's going on with blood flow now? Is the blood flow also better, or is blood flow still screwed up like before? And the answer is that the blood flow was as bad as it was before, when the animals were cognitively impaired. So with the antibody, we still have reduction in cerebral blood flow. We still have endothelial dysfunction, despite the mice having recovered cognitive function. So the data then suggests that this high salt diet leads to polarization of Th17 cells in the gut that increases IL-17, phosphorylates eNOS, leading to inhibitory activity, suppresses nitric oxide production, which in turn does two things. One, activates calpain and leads to p-tau phosphorylation tau. At the same time, reduces the blood flow, both of which could contribute theoretically to the cognitive impairment. However, because of the experiment with antibodies and because of experiments with the knockout of tau, which I didn't show you, we are able to induce normal cognition despite the fact that the flow reduction is still there. So the flow reduction at the time that the mouse is demented plays no role in the dementia, but it's tau that is driving the, the dysfunction. So to then wrap it up, concerning the role of immune factors in cognitive impairment, I showed you how innate immune cells associated with cerebral blood vessels are able to mediate the deleterious vascular effects of Alzheimer pathology by acting on innate immunity receptors like CD36, leading to production of free radicals and endothelial dysfunction, functional hyperemia, and everything else, which is really a global alteration of all factors of the pertaining to cerebral circulation, from functional hyperemia to the blood-brain barrier. So nothing works, all right? But then there is another condition that you've seen with salt, with high salt, in which you have a selective reduction of the ability of the endothelium to synthesize nitric oxide. Such reduction, as we know, is critical to unleash a tau phosphorylation, an excess of tau phosphorylation, that then leads to cognitive impairment independently of the effects of the tau on the blood flow. So the take-home message here, the lesson is that we see vascular dysfunction in a variety of conditions, but not all vascular dysfunction is the same. So the vessels can produce, can alter brain function, either by a starvation-like mechanism where there is not really, everything is not working, and like in the amyloid case, and nothing, and so the brain is at risk of getting hypoxic, or perhaps getting somewhat hypoxic. That's one way. The other way is that the brain vessels are critical for maintaining the homeostasis of neurons, all right? This is part of the neurovascular unit concept, all right? The reciprocal relationship between the two, the symbiotic relationship. And so by inducing this homeostasis in the neurons, you lead to tau hyperphosphorylation and cognitive impairment. So different mechanisms, but both vascularly related. Concerning the stroke story, we have seen now how stroke has a profound effect on systemic immunity that in turn drives a lot of the infiltration of the brain by inflammatory cells, from the innate immune cells to the adaptive cells like the T lymphocytes and so on, from the meninges, from the the skull, bone marrow playing now an increasing role, and these cells play a key role. They can damage, they can protect, and I have not shared it to you, but they also play a major role in repair, and risk factors in the microbiota play a key role in determining whether these immune factors can be detrimental or beneficial to the brain. So the immune cells are really very, very important for brain health, and both in two major neurological diseases. I'd like to thank all my colleagues and my funding sources for allowing us to do all this work. And thank you for your attention, for the invitation again.
Thank you very much, Costantino. A really stellar lecture covering tremendous ground. And I think what I'd like to do, since I invited you and I could chair a few questions from the audience, whilst they're thinking, you showed us that in the mouse, that one set of mice that had been given antibiotics where you wiped out the microbiome or certain part of the microbiome, you showed the stroke was virtually non-existent, really, really tiny. Do you think the microbiome influences, because we're in a Metallomics Symposium, the metal content of the brain? In other words, if we were to take the brains you have from those two sets of mice, would the metallomic profiling be different? Would they be less sensitive to ferroptosis? Has that ever been looked at?
Yeah, not to my knowledge. I would suspect, so the way you know, the way this kind of multifactorial neuroprotective mechanism work, I mean, and you've done a lot of work in this, is that you don't have one single thing that goes. It's more of a confluence, beneficial confluence of factors. So hypothermia, for example, affects blood flow, affects metabolism, affects oxidative stress, affects inflammation. You know, I suspect that having such a profound effect, which is a hypothermia-like effect, all right, that's 75% reduction. You gotta have to have a multifactor. I suspect that it's going to be less, you know, Fenton reaction in the brain. That it's going to be less, the other thing that in stroke now has been shown is there is increased gut permeability. So a lot of these metals, a lot of the stuff you were talking about is going to get into the circulation, and with an altered blood-brain barrier, you know, with this new meningeal, you know, skull bone marrow, you know, that's another thing that is really amazing. There are channels that go from the skull to the meninges. So in other words, whatever is in your skull, it's going to kind of drip down on your brain, and you know, and so everything is up in the air. I mean, now, I mean, and the brain becomes exposed, you know, to this shower of systemic factors. Metals may be involved, and certainly the oxidative aspects of that. Yeah.
Right. Thank you. Do any of the other speakers that are with us have questions for Costantino? Alternatively, those in the audience can place them in the chat and we can relay them on to Costantino. Richard, do you have a query, thought?
Yes, I do. It's in the chat.
I haven't looked, sorry. In the chat or in the questions?
Q&A.
Q&A, I'm looking. Okay. Published. Is there evidence that oxidative stress or phosphorylated tau amyloid beta leads to eNOS uncoupling to exacerbate brain microvascular dysfunction?
Yeah, so we did not look specifically to eNOS uncoupling, but we know that the radicals, you know, there was no increased radical formation. So eNOS, as you know, eNOS uncoupling is the mechanism when there is a starvation of substrates. eNOS, instead of making nitric oxide, makes reactive oxygen species. It's been very well described in diabetes, for example, that, you know, at least, you know, David Harrison's and so on, that there is this production of you know, kind of almost like instead of becoming a nitric oxide producer, becomes a reactive oxygen. We have no evidence that that is occurring. Actually, the activity of the enzyme is reduced, indicating that, you know, even, you know, in any case, the OXPHOS part of the NO production is also attenuated. So I doubt that that's going to be a factor.
I have here a second question from Ian Chambers, BNI with Raj, curious about the different vascular impairment models and the implications on circulating red blood cells, particularly the obstruction. Will this not cause lysis, increased recycling? Have you looked at the spleen as a source of the immune response as well?
We have not, but there is an extensive literature on the spleen.
Yes.
That shows, so essentially after stroke, as part, and that's been shown in humans too, as part of the immunosuppression, there's massive atrophy of the spleen. And that is thought to be one of the factors that contribute to the pneumonias and all of the complication, inflammatory complication after stroke. So in many models, you know, splenectomy can modulate in some models, not very many models have been tried, but in some models, you know, splenectomy can modulate the impact of any you know, injury on the brain. Again, raising the importance of this critical role of the immune system in the brain.
And finally, the last question in this one set of questions: is it possible that bacteria are thriving on the released free iron and heme systemically?
Most likely, I mean, from the red blood cells, lysis, and so on. Yeah. Obviously, human stroke is not, is not caused by inserting a filament into the carotid artery, right?
Exactly.
So we are, we are here a little bit on a shaky model. We don't want to get into the controversy about the stroke models, but certainly the people think that bacteria are getting into the brain. I mean, there is a very reputable laboratory thing, public with papers published in top-notch journals that bacteria in Alzheimer's disease, A beta may be an antibacterial agent that is protecting the brain from bacteria. So iron is vital to as you teach me, vital to the bacterial, life, and so wouldn't be surprised.
Thank you. I don't see any more questions. So on behalf of the London Metallomics facility, I'd like to thank you in particular as our keynote speaker, Costantino, and all the other lecturers who participated, and certainly the audience. And then I'd like to give a big thank you to our IT team and hand over for the final summary to Dr. Richard Xiao.
Thank you, Giovanni. Thank you all.
Thank you, Giovanni, especially to Costantino. That was a wonderful presentation, a real tour de force, very enjoyable. And of course, all of the other speakers this afternoon. I'll keep my conclusion very brief because many of us have been here for over 5 hours and glued to our seats with this exciting symposium. But I would also like to mention our very kind sponsors who have supported not only this symposium, but also the London Metallomics facility over the past couple of years. And I don't know if you can see this slide. If you can let me know if the slide is
visible.
Shown.
Yes, we can see it, Richard.
So these are our sponsors. On the left-hand side, really supporting the London Metallomics facility, Teledyne, Thermo Fisher, and of course Unilever. And on the right-hand side, Baker Ruskin and BMG Labtech have been very kind to support the physiological oxygen laboratory here at King's. So thank you again to our sponsors. So if I stop sharing my slide, and just to again thank the audience. I know there are only 45 of you who are remaining, but we did have a very large audience at the beginning. Thank you so much for joining us wherever you are. This is not only in London, but we have attendees globally. So thank you again for joining us. And of course, to the speakers, the organizers, the LMF team, thank you for making this such a wonderful event this year. And we also welcome you to have a look at our websites for the London Metallomics facility. Many of you would have seen it already. And I will post the links to our pages, our websites in the chat. So thank you again to all of you for joining us this afternoon.